Welcome to the cancer biology course, and this is the cancer biology course for the beginners, and it covers all the topics for an advanced learner. My name is Diptano, and I'm a PhD research scholar in computational biology, and I'm also the founder of DHelix Bio. Uh, so, talking about the course, we'll be looking into the different uh, concepts, and we'll be starting with the introduction, and this will follow the molecular mechanisms and then the cellular biological foundations. For example, we'll be looking at the cell cycle processes in cancer biology, the hallmarks of the cancer, and then the genetics and the epigenetics, genomics of the cancer, the transcriptomics, epigenetics, and the regulation in the tumors. This will be followed by the tumor microenvironment and immune contexture, and this will also include the tumor evolution and some parts of the cancer diagnostics and the biomarkers. It will also cover the immune context of the cancer development, certain therapies from the targeted drug therapies, targeted drug development and the immunotherapies, and also CAR T conceptually, uh, checkpoint inhibitors, and then resistance mechanisms, and like we'll also cover the preclinical models, translational pipelines, and computational cancer biology, like the different databases and different data sets, as well as the different programming languages which will be used to analyze these data sets. We'll also cover the multiomics integration of the network biology, and followed by the clinical trials and the regulatory concerns and ethical concerns. Then we'll have a glance of the data science and artificial intelligence in oncology, and a fewer case studies on different uh, cancer profiles. So, introducing with the cancer biology. So, as we have already studied about cancer, there are a lot many videos and the tutorials and a lot of the things. So, basically, cancer is uncontrolled growth of the cells. So, when the normal cells basically grows uncontrollably, this causes a disease called cancer, and it is acquired by the genetic and epigenetic changes, which we'll be looking in the upcoming slides. So the normal cells basically ignore the normal regulatory signals, and then they turn into the cancer cells, and they have another characteristics that they invade the surrounding tissues and metastasize within the distant organs. So the normal cells and the cancer cells are different in the image that you can see here, that the normal cells are comparatively having larger cytoplasm, whereas the cytoplasm size is reduced because of the larger nucleus and the shape of the nucleus and multiple nucleoli. So the chromatin is fine, and there is single nucleus in the normal cells, whereas there are multiple nucleoli, and a coarse chromatin in cancer cells. So talking about the statistics, tobacco smoking ranks the highest in the uh, increasing cancer risk in contributing in the contribution of the cancer, followed by the excess body weight or the obesity. Then comes the alcohol consumption, which is followed by the ultraviolet radiation exposure due to the sun's ultraviolet UV rays, and again poor diet is there, which leads to the excessive body weight or the overweight, and pathogenic infections like the human papilloma virus, HPV, or HIV viruses. All these can also cause the cancers, and the physical inactivity is also responsible for causing the cancer. So common avoidable top carcinogens are tobacco, areca nut, or the betel nut. Then you have the alcohol. Then air pollution, like with particles of less than 2.5 microns, can be responsible for cancers, for lung cancer especially. Then there are the viral infections, then coke production, and radioactive agents. Sorry, and we have the dust and iron ore mining. These are all top carcinogens which have been recorded globally. So, looking at the statistics, like diabetes is 2%, whereas 7% is responsible. Cancers are responsible for 7% of all the global death and diseases. Others are, you can check in this graph, where lung disease, heart disease, and other non-communicable diseases contributes to the risk. So the top 10 most deadly cancers are both lung cancer are both in men and women globally and in India, whereas in Indian women the breast cancer is more prevalent, and there are more different other types of the cancers which you can see in these graphs. So classification of the cancers by the biological behavior. So we start with the central regulatory term called the neoplasm, which is basically the first stage of the cancers, where the normal cells turn into the constantly growing mass of the tissues, and the forming a mass and forming the mass it results into the tumor. So neoplasms can be of the different types. This can be benign neoplasms, in situ neoplasms, and malignant neoplasms. Benign neoplasms are the centrally located and localized, have their localized growth. They don't invade the surrounding tissues, do not metastasize. The cells are well differentiated and resemble the tissue of the origin. They're usually slow growing and often encapsulated. For example, the adenoma, lipoma, and fibroma are some of the benign neoplasms. This can be curable and can be curable by surgical removal. uh, uh, Talking about the in situ neoplasm, they are the intermediate neoplasms or the borderline neoplasms, which are have the characteristics of the local invasions. They are limited, but they have no metastatic potential as well. Their behavior is unpredictable and often described as the locally aggressive. So then comes the borderline ovarian tumors. Uh, so basically this is one of the in situ neoplasm. So then comes the malignant neoplasms, like invasive growth, destruction of the adjacent tissues are some of the characteristics of the malignant neoplasm. They have the metastatic potential, which means that they can spread via the lymphatics or the bloods and the body cavities, and have the variable differentiation. So they may also show a marked atypia, which we'll be learning in the coming slides, and often marked by the rapid growth with the genetic instability. They can be life-threatening and require multimodal treatments. So, talking about the classification of the tumor or the cancers by the tissue of origin. So, basically, neoplasms can be classified into different types, in which there is carcinomas, there is sarcomas, there is hematologic neoplasms, there is CNS neoplasms, which is related to the brain cells or the neural cells, and there is the germ cell tumors. So carcinoma are the cancers of the epithelial cells, which is basically the surface of the glands or the surface of the, like for example, skin, and they can spread via the lymphatics. So basically, adenocarcinoma is one of the examples of carcinoma, followed by the squamous cell carcinoma and the translational cell carcinoma. There are sarcomas. Sarcomas arise from the connective tissues, for example, the bones, muscles, and the fat and the blood vessels. For example, they are the osteosarcomas, liposarcoma. Liposarcoma is the sarcomas from the fats, and osteo is from the bones. And they are less common but very aggressive, and they can spread mainly through the bloodstream. Then there comes the hematologic neoplasms. In this, there are two types of the hematological neoplasms, which are the leukemia and the lymphomas. So leukemia are the malignancies of the blood forming cells, and no solid tumor mass and malignant cells are observed. And the malignant cells circulate in the blood, and the bone marrow failure is very common. And lymphomas are the solid tumors of the lymphoid tissues, and they can be present as lymph nodes or extranodal nodes. Then comes the neuroectodermal neoplasm or the CNS neoplasms, which are derived from the neural or the glial cells, which is basically the brain cells, and they cause the disease by local invasion and increased intracranial pressure. There comes the germ cell tumors, which originate from the germ cells and occurs in the gonads or the midline sites. Some of the examples are the seminoma, teratoma, yolk sac, and yolk sac tumor. So before we proceed, we need to understand some of the terms and the definitions and how they relate to the next step. So basically, aplasia is a complete absence of the organ, which is due to the failure of the development of the tissue or the complete organ. So if it is partial development occurs, then the hypoplasia may result. So what is hypoplasia? It's the condition with the below average number of the cells leading to the underdeveloped organs, and as a result, the surviving cells may later undergo the adaptive changes. Then comes the atrophy. This reduction in the organ size due to the decrease in the cell size or maybe number as well, but mostly due to the cell size with reduced function. So a reversible adaptive response if the stimulus is removed. So hypertrophy is the increase in the cell size leading to the organ enlargement and the no new cells are formed. They are often accompanies the hyperplasia in the tissue capable of the division. So hyperplasia is the increase in the number of the cells due to the uncontrolled and regulated proliferation. And as a result, the persistent stimulation can push the cells towards the abnormal phenotypes. Now we know about the metaplasia, the reversible conversion of one differentiated cell type to another for better suited to stress. This prolonged stress may induce the dysplasia. The prosoplasia is the development of the new or the specialized cell function during the differentiation, and it represents the normal or adaptive differentiation opposite to the anaplasia. Now, dysplasia is the disordered growth and the loss of the cell uniformity and architectural orientation, and considered as a pre-malignant state. Neoplasia are the uncontrolled anonymous proliferation of the cells and persisting after the stimulus removal, and neoplasms may be benign or the malignant. So benign tumors are the neoplasms with no invasion or the metastasis and is usually well differentiated. However, they can progress to malignant forms in some tissues, which results the desmoplasia, the reactive fibers connective tissues growth induced by invading tumor cells and are commonly associated with the malignant tumors. So malignant tumors are the neoplasms with the invasion, metastasis and the loss of the differentiation. They are defined by the anaplasia and aggressive behavior. Anaplasia is the loss of the structural and the functional differentiation within the cells, and they are the hallmarks of the malignancy. Pleomorphism and nuclear atypia is the variation in the cell size or the shape with abnormal nuclei and increased mitosis. So morphologic evidences of the anaplasia, they are the morphological evidences of the anaplasia. Carcinoma are the malignant tumor of the epithelial origin, example the squamous cell carcinoma and the adenocarcinoma. One of the major class of the malignant neoplasm are carcinoma. Sarcomas are the malignant tumor and mesenchymal origin. For example, the fibrosarcoma, liposarcoma, and they are aggressive and hematogenously spread. Sarcomas are the malignant tumors of the. We just talked about the sarcoma. So teratoma is the neoplasm which contains the tissue from more than one germ layer and demonstrates the disordered differentiation. Then comes the oncology, which is basically the integration of all the process, and it's the scientific and the clinical study of the tumor and the cancer biology. So in this slide, you can see that uh, all the previous terms that we have studied earlier are related here. So suppose there is a complete failure of the development of the organ. So this condition is called aplasia. Now hypoplasia is the condition in which the cell number is reduced. So as a result, the size of the organ will also be reduced. If the cell numbers are increased, the size of the organ will be increased, and this phenomenon is called the hyperplasia. On the contrary, if the cell size is reduced, the organ can also get reduced, and this phenomenon is called atrophy. Whereas hypertrophy is the condition in which the cell size is enlarged, and as a result, the organ size can also get enlarged. Now, the normal cell can basically transform into different types. One is the metaplasia, which is into different type of the cell. Prosoplasia is into uh, it develops some different function, whereas in dysplasia there can be different functions as well as loss of the cellular uniformity and the function. So basically, dysplasia can give rise to the neoplasia, which means they are in the early stage of the development of the cancer or the tumor, which can then give rise to the benign tumor. And as a result of anaplasia, the metastasis can happen and malignant tumors can form. So basically, this is the image of the lung cancer. So basically, the suppose the liver cancer can travel and they can contribute metastasize into the lung cancer. So carcinoma are, as we have studied earlier, they are the uh, cancers of the squamous cell, and they contribute with single kind of the cells, whereas the teratoma are the cancers with multiple uh, multiple types, different types of the cells. So now we come to the hallmarks of cancer. So there are 14 different hallmarks of cancer, which have been collected from, so these 14 hallmarks have been collected from a review paper, and these are known as these are known as the hallmarks of cancer because they show the distinct characteristics in the development of the cancer. So the first hallmark of the cancer is the sustaining proliferative signaling. So we can see this in comparing them in the normal cell as well as the cancer cell. In normal cell, what happens is the normal cell grows when there is an external signaling or external stimuli called the growth factor, and the growth factor can bind to the some receptors like receptor tyrosine kinase, and as a result these pathways like MAP kinase and the PI3K or the phosphate inositol 3 kinase, these kinds of pathways are activated and they control the proliferation. However, the normal cell's growth requires the external signalings, and these external signalings are called the growth factors. In cancer, what happens, these growth factors are synthesized by the cancer cells themselves. So basically, these cancer cells basically keep on constantly signaling the growth factors, and they have the same receptors, which again carries on the different pathways like MAP kinase and PI3K, AKT, and mTOR, mammalian target of rapamycin pathways. mTOR pathway results in the protein synthesis and the survival, whereas the MAP kinase gives the cyclin transcription. These persistent cyclin D continues the G1 to S phase entry of the cell cycle from G1 to S phase, which results in the synthesis of the necessary proteins and like uh, uh, makes the cell compatible to grow further, to regenerate. Sorry, differentiate. Now, the therapeutic blockade can be performed by if we can target these receptors like the receptor tyrosine kinase or mutated receptors like EGFR and HER2, and certain inhibitors or antibodies can act on these receptor surfaces to block them. So, in this way, the cancer cells can be stopped with the autonomous growth. Now, talking about the second hallmark of the cancer is the evading growth suppressors. So basically, there are certain growth suppressors like RB protein, or the which are basically which maintains the RB pathway. So basically, there are the cyclin D and the CDK4/6. They basically are inhibited by the P16, and as a result, the RB protein can bind through the E2F, and this acts as a checkpoint for the G1 or the S phase of the cell cycle, as well as the checkpoint for the G2 or the M phase of the cell cycle. And as a result, this protein can stop the growth of the or the suppress the growth of the cells further. There are certain conditions in which, like for example, the DNA damage, in which the P53 pathway is activated, and activated P53 protein can inhibit the P21, which is again the inhibitor for the apoptosis, and as a result, the apoptosis and the DNA repair, this normal cells stops the growth. Apoptosis leads to the cell death, and the DNA damage repair leads to the damage of the damaged leads to the repairing of the damaged DNA. Whereas in contrary, the cancer cells have the evading growth suppressors, which means they can evade the growth suppressors. For example, the RB pathway, in which the cyclin D and the 4/6, they basically are. So basically, the phosphorylation of the RB can result into the E2F, release of the activation of the E2F, which basically enters the S phase and enhances the proliferation. And in this case, the G2 or the M phase checkpoint is bypassed. Similarly, in the P53 pathway, where the DNA damage can result the mutated P53, which basically inactivates, and the apoptosis and the DNA repair is not proceeded. So the therapeutic in the therapeutic angle, we can see that we can re-engage this checkpoint control by use of the CDK4 or 6 inhibitors, which prevent the phosphorylation of the uh, RB protein or the retinoblastoma protein. The third hallmark is the resisting cell death. So in normal cells, apoptosis is triggered, which means the normal cells create a death signal, and there are certain proteins due to the DNA damage called the pro-apoptotic proteins. So after the pro-apoptotic proteins, they will trigger the uh, apoptosis um, via the MOMP of the mitochondria. The MOMP will release the certain proteins called the cytochrome C, which will activate the caspases, and as a result, there will be programmed cell death. Whereas on cancer cells, the apoptosis is evaded. The death signals when they reach the cells. So basically, the cancer cells overexpresses the anti-apoptotic proteins called the BCL2, which basically masks the death signals, and they never reach the mitochondria. And as a result, the MOMP is suppressed, which basically inhibits the caspase activation, and as a result the cells accumulate the mutations without being eliminated. So the therapeutic logic is that you can inject the person with BCL2 inhibitor, which is basically an anti-apoptotic BCL2 inhibitor, and it inhibits the protein called BCL2. As a result, the MOMP is basically activated, and this forces the apoptosis by caspase activation, and this is the therapeutic translational angle for this hallmark. The fourth hallmark is the enabling the replicative immortality. So basically, cancer cells have unlimited divisions, and they can replicate immortal in immortal manner. So in normal cells, there is the limited divisions. So this is because of the telomerases. So the telomerases basically keep on shortening on every divisions. On every successive divisions, the telomerase shortens, and they are the basically the small caps at the end of the telomeres. So these the shortening of these telomerases can result in the senescence or the cell death. Whereas in the cancer cells, the enzyme TERT reactivates these telomerase enzymes, and basically they indefinitely elongate the telomerases, and as a result, the alternative length of the telomerase ALT via recombination is activated, and as a result, this avoids the senescence and the crisis. The fifth hallmark of the cancer is the inducing or accessing vasculature. So basically, this hallmark explains that the cancer cells are very hyperactive, and they are constantly looking for energy and in the form of the oxygen, and as well as the nutrients. But the diffusion limit is 1 to 2 millimeter, and as a result, there is a crisis of the oxygen in the cancer cells. Therefore, there is a protein which is called HIF1 alpha, and this basically is activated, and it results in the formation of the several other proteins called the VEGF, PDGF, and angiopoietins. So basically, what they do is they sends the signals for endothelial proliferation and sprouting of the vessels. So basically, new blood vessels are formed, which is termed as the angiogenesis. And as a result, abnormal leaky tumor vasculature are promoted, and this enhances the nutrient and oxygen delivery in the cancer cells. The other hallmark is the activating the invasion and the metastasis. So in primary tumor, the epithelial mesenchymal transition happens. So EMT happens, which is basically the detachment. So not all the tumor cells may survive, but those which survive, they can detach from the existing tumor. And by the activation of the MMP or the matrix metalloproteins, they can travel through the blood vessels, and which is called the intravasation, which is basically they can go into the blood vessels of the blood streams, and they can travel to the other parts of the uh, body and the distant organs like the lungs and the bone. So intravasation is basically these cells enter the bloodstream, and extravasation is they exit the bloodstream and metastasize and colonize a new organ or the new region of the body. So the the seventh hallmark is the deregulation of the cellular metabolism. So basically, in normal cells, there is efficient metabolism. So the glucose is basically transported by the GLUT1, and the normal glycolysis happens, which basically produces less amount of ATP. But this is further followed by the mitochondrial Krebs cycle, and this produces a large amount of ATP as well as carbon dioxide. So this is how the normal cells efficiently produce the ATP. However, in cancer metabolism, there is an Warburg effect. So basically, this is the aerobic glycolysis. Similarly, the glucose will enter the GLUT1, and it will enter in the large amount. The intake of the glucose is enhanced in cancerous condition, and as a result, there could be a partial degradation and aerobic glycolysis, and the mitochondrial Krebs cycle is also there. But this doesn't limits itself to there. It instead keeps on producing other necessary compounds for uh, cell differentiation. For example, the synthesis of the nucleotide, lipid synthesis, and amino acid synthesis. So the cancer cells basically rely on the rapid biomass accumulation, and as a result, the lactic acid is also produced, and which gives an acidic microenvironment and aiding the invasion. And the way we can restrict is by using the glycolysis inhibitors. So basically, this is a therapeutic angle, and exploit the metabolic dependencies. Coming to the other hallmark of the cancer is the avoiding immune destruction. So basically, cancer cells avoid the immune destruction. In normal cell, there is effective immune response where the tumor are eliminated because the normal cells presents the antigens, and these antigens fragments basically bind to the T cells, and as a result, there are certain cytotoxins are produced, and these cytotoxins basically break down or eliminate the tumor cells, and this process is called the immune system, which destroys the tumor cells. But this immune system is evaded in the cancer cells, and they bypass this thing. So basically, the cancer cells will secrete certain proteins like PDL1 and PD1. These T cell exhaustion are due to the PDL1 and the PD1 interaction. So basically, they will bind to these cancer cells, and the cytotoxins are not released. And as a result, the regulatory T cells, or the Tregs and MDSCs, and they recruit the inhibitory cells, and as a result, there will be loss of the antigen presentation, and the immune system fails to eliminate the tumor cells. So the therapeutic angle is that certain inhibitors of the PD1, PDL1 inhibitors, they can bind to these surface antigens, and as a result, the cytotoxins will be normally produced, and this will create the checkpoint for inhibition and reactivation of the immune response. Coming to the next hallmark is the tumor promoting inflammation. So there is chronic inflammation, which is basically the in the tumor promoting environment, and the chronic inflammation can create a mutagenic and the growth promoting environment, basically by the secretion of the reactive oxygen species, and which causes the damage to the DNA. So certain cytokines like IL-6 and the TNF alpha can basically bind to the tumor cells, and they can activate the STAT3 and the NFKB. This is followed by the macrophage derived growth factors, and as a result, increased mutation rate and enhanced survival and proliferation may occur. The another hallmark of the cancer is the genome instability and the mutation. Whereas in normal cells there is efficient DNA repair. Suppose there is the repair in the BRCA enzyme, BRCA gene, BRCA1 gene, or breast cancer gene, and as a result there will be a pathway for the repair of the this gene or the DNA, like the BRCA repair pathway, and as a result there will be successful repair in the normal cells. However, in the cancer cell, there is a defective. These proteins are get defected. The defective DNA repair or BRCA mutations, and as a result, there will be chromosomal instability and the accelerated mutations, accumulation of the accelerated mutation. One therapeutic principle of this is the use of the PARP inhibitor, which basically binds to the DNA and exploits the repair defects. And by inhibiting the BRCA mutant cancers, and the cell death is initiated. The This is followed by the hallmark, which is called the unlocking the phenotypic plasticity. So basically, the primary tumor are in the epithelial state, and with the help of this TGF beta, WNT, and EMT programs, which are activated, the cell state is switched, and there will be dislocation of the of this endothelial mesenchymal transition, and they will be basically evading the new places, new organs or distant organs, and this is called the MET during uh, uh, colonization, and this initiates the metastasis, and this is called the distant colonization and mesenchymal to epithelial transition, and so this is like the acquisition of the stem-like properties is observed in these kinds of cancer cells. So basically, they act as the stem cells, and they can colonize the distant organs. So then comes the epigenetic reprogramming. So basically, in normal cells, the gene expression happens when the chromatin, which is basically their tightly regulated in the histones, but with the minimal DNA methylation and the acetylation of the histone proteins, there can be damage of the DNA, and as a result, there can be the formation of the mRNA, which are basically the differentiation genes, and they become active. However, in the cancer cells, the epigenetic reprogramming and the gene expression is rewired, where the histone modification and the changes and the chromatin remodeling can silence the differentiation of the genes, and as a result, there will be DNA hypermethylation with enhanced plasticity. So, the other hallmark of the cancer is the non-mutational epigenetic instability, whereas the therapy response or the transient state is observed when the drug molecules affect the cancer cells. So basically, there is the non-mutagenic epigenetic instability, and basically the reverse resistance. So the drug tolerance is persistent in both the states, and as a result, there will be stress-induced chromatin changes, and the transient transcriptional states allow the survival under the therapy. The clinical significance is that the resensitization strategies happens, and this explains the relapse without the new mutations. The final hallmark of the cancer is the polymorphic microbiomes. So basically, there are the healthy microbiomes, and basically there are the dysbiotic microbiome. So healthy microbiomes help in the immunotherapy, and they have the strong immune response, and they act as anti-tumor agents, whereas they can convert into the polymorphic microbiomes, and they resist the immunotherapy, and as a result there is a weakened immune response, and this is basically pro-inflammation and the altered response. So the molecular mechanisms of cancer are that there are certain genetic alterations and the DNA repair defects, and as a result, the point mutations, indels, and translocations may occur, which basically follows the chromosomal changes, and there is a defect in the DNA repair machinery. This gives rise to the genomic instability. Similarly, there is the cell cycle dysregulation, where the cyclin and the CDK overactivation may inactivate the P53, RB proteins or retinoblastoma proteins, and the loss of the P53 may occur. These are basically the checkpoints in the cell cycle. So once the loss or the inactivation of these proteins may result into the uncontrolled G1 or the S transition, resulting into the autonomous proliferation. This is followed by the epigenetic dysregulation, which is basically non-mutational due to the DNA methylation. The histone modifications may occur, and this can silence this can cause the silencing of the differentiation and the chromatin remodeling defects. And this is followed by the deregulated signaling and the apoptosis evasion. And as a consequence, there is an uncontrolled growth and adaptability. Now let's look at how the cell cycle is altered in the cancer. So basically, we know that mother cells give rise to the daughter cells, and this is the two phase in the cell cycle. One is the interphase, and the other is the mitosis or the M phase. So in interphase, there is the G1 phase, where the mother cells grow in the size and produces the RNA and proteins and prepares for the DNA replication. At the end of the G1, it basically enlarges, the size is enlarged, and then it comes to the S phase or the synthesis phase, where the DNA is replicated so that each daughter cells will receive an identical genome. This is followed by the G2 phase, where there the further growth occurs and the cells check for the DNA damage before division. And this is followed by the mitosis. So there are the four different phases in the mitosis. This is the prophase, metaphase, anaphase, and the telophase, which is again the cell biology context. And we have the cytokinesis, and cytokinesis gives rise to the so basically cytokinesis is the division of the two cells, and the two daughter cells will be produced. Now there are certain critical checkpoints, and these are called as like spindle checkpoints, and there are certain regulatory proteins associated with them, and these are the CDKs, tumor suppressors, for example the P53 and the DNA repair enzymes. Now let's look into the regulatory proteins that control the. So in this slide, we are looking at the regulatory proteins that control the cell cycle, which ensures that the cell divide in a controlled and orderly manner. So at the center of the regulatory regulation, there are the two key components. These are the cyclins and the cyclin-dependent kinase, or the CTKs. So CDKs are the enzymes which specifically catalytic kinase, but they are inactive on their own. They only become active when they bind to appropriate cyclin. And on the other hand, the cyclins are regulatory proteins whose levels fluctuate throughout the cell cycle, and they synthesize and degrade at the specific stages. When the cyclins bind to the CDKs, they form the cyclin dependent kinase complex, or the cyclin CDK complex, and these complexes function by phosphorylating the target proteins, which drives the cells forward from one phase of the cell cycle to the next, so progressing from the G1 to the S phase, and then to the G2, and finally into the mitosis. So during the G1 phase, the cyclin D binds to the CDK4 and the CDK6, promoting the gene expression and helping the cells to prepare for the DNA replication. As the cell approaches the G1 to the S phase, the cyclin E binds to the CDK2, which is crucial for the initiation of the entry into the S phase. Once inside the S phase, the cyclin A associates with the CDK2 and regulates the DNA replication and ensures that the genome is activated in response to the DNA damage and can induce the cell cycle to arrest. As the cells progress towards the G2 and prepares for mitosis, the cyclin A and later the cyclin B interacts with the CDK1, ultimately triggering the event of the mitosis. However, this process is not unchecked. The cell cycle is tightly regulated by the inhibitory mechanisms, and these are the CDK inhibitors such as the P21, P27, and P16. They can bind to the cyclin dependent kinase complexes and block their activity, effectively halting the cell cycle when necessary. Tumor suppressor proteins also play a critical role. For example, the P53, often called the guardian of the genome, and it is activated in response to the DNA damage and can induce the cell cycle arrest. Uh, Another important protein is the RB protein, or the retinoblastoma protein, which regulates the gene expression and controls the transition from the G1 to the S phase, and this prevents the premature progression. In addition to this, the cell has the checkpoint sensor systems, like the proteins like ATM and the ATR, that detects the DNA damage, and they activate the downstream effectors such as the CHK1, CHK2, which basically enforces the cell cycle arrest until the DNA damage is repaired. Finally, the entire system is influenced by the external and the internal conditions, such as the nutrient availability, then cellular stress, DNA damage, and the differentiation signals. And these inputs ensures that the cells only divide when the conditions are favorable and the genome is intact. This slide explains the DNA damage response pathway in the normal cells and shows how the cell cycle is tightly controlled and how the cells respond to the DNA damage before proceeding the divisions. The process begins with the resting cell in the G0 and the early G1 phase, and at this stage the cells receive the growth signals from the environment, and these signals are evaluated at a critical checkpoint known as the restriction point or the R point, and only if the sufficient growth signals are present, they do the cells commit to ensure the cell cycle. At this point, the protein RB or the retinoblastoma protein plays a critical role in its unphosphorylated state. RB acts as a brake and prevents the cell proliferation. When the growth signals accumulate, they activate the cyclin D1 CDK4 complex or the CDK6 complex, which phosphorylates the RB protein. And as a result, this phosphorylation inactivates the RB, releasing its inhibitory effect and allowing the cells to commit the proliferation. Following this, the cells progress through the G1 with the help of the cyclin E and the CDK2 complex, and which drives the transition from the G1 to S phase. And during the S phase, the cyclin A and the CDK complex ensures that there is a proper DNA synthesis and coordination of the replication. As the cell approaches the mitosis, the cyclin B and the CDC25 and the CDK1 complex they become active, triggering the entry into the mitosis by removing the inhibitory phosphates from the mitotic CDKs. And a proper regulation ensures that mitosis proceeds correctly, and exit from the mitosis involves degradation of the cyclin B and inactivates the CDC25. At multiple stages, the cell uses the internal checkpoints to verify the genome instability and ensures that the earlier steps have been completed correctly. If the DNA damage occurs, it activates the tumor suppressor or the p53 protein, and activated p53 induces the expression of the CDK inhibitors such as the p21 and p27 and which blocks the cyclin CDK activity and halt the cell cycle. Additionally, the signals like the TGF beta can reinforce the inhibition. Other regulatory proteins, such as the ATM and the ATR, detect the DNA damage, and through the downstream signaling pathways, they activate the proteins like the CHK1 and CHK2 to enforce the cell cycle arrest. If these regulatory mechanisms are functioning properly, the cells either repair the damage before continuing and preventing the damaged cells from dividing. However, this slide also highlights the loss of the mutation, and these regulatory proteins removes these restraints, allowing the cells with the DNA damage to continue dividing, which is a key step towards the cancer development. So this slide compares the normal cell cycle with the cancer cell cycle and highlights how the regulation is lost during the cancer development. In normal cells, the cell division is tightly regulated by the checkpoints and signaling pathways. These checkpoints ensures the cells only progresses when the conditions are appropriate. And in contrast, the cancer cells, this regulation is poorly controlled, and the checkpoints are often bypassed or completely disabled, and allows the continuous divisions. Normal cells respond strictly to the growth signals, and they divide only when the cells receive the appropriate external signals from the environment. However, the cancer cells can divide without these external signals, meaning they become independent of the normal growth and control mechanisms. The checkpoint function is another key difference in the normal cell, where G1 and the S and as well as G2 and the M checkpoints are functional, preventing the cells from the damaged DNA from progressing further. Whereas in cancer cells, these checkpoints are defective or ignored, and it allows the damaged DNA to pass on during the division. When the DNA damage occurs in the normal cell, it triggers the repair mechanism or the apoptosis and ensures that defective cells do not survive. And in cancer cells, the DNA damage is often ignored, leading to the accumulation of the mutations over time. The tumor suppressor genes such as the p53 and the RB plays an important role in normal cells by inhibiting the abnormal cell divisions, and in cancer cells these tumor suppressors are often mutated or inactivated, removing an important layer of control. The rate of the cell division is also very different. The normal cells divide in controlled and limited manner, whereas the cancer cells divide very rapidly and uncontrollably. Another important feature is the contact inhibition. Normal cells stop dividing when they come in contact with neighboring cells, maintaining proper tissue structure. And the cancer cells lose this property and show no contact inhibition, which causes them to pile up and form tumors. Genetic stability is maintained in normal cells where the genome remains relatively stable over time. And in contrast, the cancer cells exhibit the high genomic instability, leading to the frequent mutations and chromosomal abnormalities. Finally, in normal cells, the damaged or the abnormal cells undergo apoptosis or the programmed cell death, which acts as the protective mechanism in cancers. And apoptosis is often avoided, allowing the abnormal cells to survive and continue dividing. This brings us to the next slide, which is genetic and the genomic instability. So genetic instability refers to the set of the events which are capable of causing the unscheduled chromosomal abnormalities or the smaller scale mutations in the DNA, either of a temporary or a permanent nature within the genome. This can happen in the two different levels. These are the nucleotide level and the chromosomal level. The nucleotide level is characterized by the faulty DNA repair pathways, base excision repair, nucleotide excision repair, and includes the instability of the microsatellites repeats or MSI, and it is caused by defects in the mismatch repair pathway. Whereas at the chromosomal level, it is characterized by the chromosomal alterations, the results in the gain or the losses of the whole chromosomes, inversions, deletions of the duplications. The translocations of the large chromosomal segments can be the characteristics of the chromosomal level of the genomic instability. So several pathways are implicated in the chromosomal instability. The pathways involved in the telomere and the centromere stability, then the cell cycle checkpoints pathways and the kinases, pathways regulating the diverse proteins via the translational modification, post-translational modifications, then the sister chromatic cohesions and the chromosome segregation and the centromere duplication. So genetic alterations are caused due to the different factors like base substitutions. Then there is the deletion and the insertion. And alterations in the chromosome number are caused due to the loss of the chromosome, for example, the loss of chromosome 10 in glioblastoma, which often reflects the inactivation of the tumor suppressor and the gene P10. Uh, the gain of the chromosome, for example, 7, chromosome 7 in papillary renal carcinoma, reflecting a duplication of the mutant oncogenes. So types of the genetic alterations in the tumors. The first one is the subtle sequence change or the point mutation. These are the small scale alterations involved in base substitutions or the insertion or the deletion of a few nucleotides. Although the minor in size, they can dramatically affect the protein function. For example, the missense mutations can convert the proto-oncogenes into the oncogenes by producing constitutively active proteins. A classic case is mutation of the KRAS gene, which occurs in more than 80% of the pancreatic cancers and leads to the continuous growth signaling. Such subtle mutations can be common and are uh, often evade the DNA repair mechanisms. The another type of the genetic alteration is the chromosomal rearrangement. This involves the large structural changes such as the translocation, inversion, or the deletions of the chromosome segments. So rearrangements can create the fusion of the genes with abnormal functions, and they can also place the genes under the control of the inappropriate promoters. A well-known example is the formation of the fusion oncogenes that produce the proteins with enhanced or deregulated activity. These changes are particularly common in leukemias and lymphomas. Gene amplification and copy number alterations. In this category, the certain genes are present in the multiple copies, leading to the overexpression of the growth promoting proteins. The gene amplification can occur as an extra chromosomal DNA fragment or as a repeated chromosomal regions. This increases the gene doses, enhances the proliferative signaling and survival advantages in the tumor cells. This mechanism is frequently observed in the aggressive and the solid tumors. This is followed by the aneuploidy, and which is basically the whole chromosomal changes. And aneuploidy refers to the gain or the loss of the entire chromosome. So basically, it results in the abnormal chromosome numbers, and this leads to the widespread imbalance in the gene expression, and contributes to the genomic instability. The aneuploid cells are more prone to accumulating the further mutations, accelerating the tumor progression and the heterogeneity. So here you can see the images of the inversion, deletion, and the insertion. Where in inversion, the the amino acid sequences are inverted. So AGCC is followed by TCGG, and vice versa. Deletions means the complete removal of the of certain amino acids from the DNA, and insertion means the some new genes, some new amino acids are some new nucleotides are inserted. Similarly, the translocation is can be of the different types. So basically, there will be translocation of suppose this B and A, they are translocated into the other chromosome, whereas the KJI, they will be translocated to the adjacent chromosome. So basically, this is called the translocation. So molecular mechanisms which maintains the DNA fidelity. So basically the newly synthesized DNA contains the nucleotide mismatch. So there can be some mismatch of the nucleotides. So DNA polymerase will recognize these mismatch base pairs, and the exonuclease is used to cut the incorrect nucleotides, and this is followed by the correct nucleotides that will be replaced, and as a result the process of the DNA replication continues. So basically, there are the three different types of DNA fidelity mechanisms. These are the NER, MMR, and the BER. So basically, the nucleotide excision repair, DNA mismatch repair, and the base excision repair. So talking about all of them in details. So nucleotide excision repair, it's a particularly important in mechanisms that removes the DNA damage induced by the ultraviolet radiations. So you can see that in the nucleotide excision repair, so the DNA damage is caused by the radiations, and as a result there will be two different types of the repair. There will be transcription coupled repair and the global excision repair. And this damage results in the bulky DNA adducts, and these adducts are mostly thymine dimers or the 6,4 photo products. The recognition of the DNA damage leads to the removal of the short single-stranded DNA segments and contains the lesions. This undamaged single-stranded DNA remains, and DNA polymerase uses it as the template synthesize a short complementary sequence. So the DNA damage is recognized, and this will be followed by the ligation of the ligation to complete the nucleotide excision repair, forming a double-stranded DNA, and is carried out by the DNA ligase. So nucleotide excision repair can be divided into two subpathways. This is a global genomic NER or the transcription coupled NER. The two subpathways differ in how they recognize the DNA damage, but they share the same process for the lesion incision repair and ligation. So in this way, you can see that in this image, the DNA damage is recognized, and several factors will come and bind with this damaged part, and this will unwind and repair uh, the damaged parts, and the DNA polymerase fills the gaps and seals the nick, and creating a normal nucleotide sequence, which is basically restored after the repair of the nucleotides. This can be in two different types. This can be clean template for the transcription and the clean template for replication. So the second type is the DNA mismatch repair or the MMR, and in this despite of the proofreading, the DNA polymerase occasionally insert the wrong bases or slip while copying repetitive sequences and creates the base mismatches from the earlier proofreadings, and these errors are corrected by the mismatch repair or MMR system, which basically acts as the DNA replication and is uniquely strand specific. The repair machinery identifies the newly synthesized daughter strand and selectively removes the incorrect segments. And importantly, this does not excise just a single base, instead removes a stretch of the DNA and can range from the few nucleotides to more than thousands of the base pair. So basically the DNA polymerase acts on these nucleotides, and as a result MMR intact of the 6 repeats here and the MMR deficiency of the 7 repeats here. This basically corrects this, repairs these mismatches. And in mammalian cells, these mismatches are first recognized by the two protein complexes. This is the HMSH2, HMSH6 complex, and which detects the base-base mismatch and the small loops, while the another complex, which is called HMSH2 and MSH3, uh, mainly recognizes the insertion deletion loops, and these complexes then recruit the HMLH1 and the HPMS2 and forms a repair assembly that attracts the additional factors such as the exonuclease, DNA polymerases, and ligases in order to remove and resynthesize the faulty DNA. By doing this, MMR increases the accuracy of the DNA replication by 100 to 1000 folds and reduces the error rate to approximately one mistake per base pair per 10 to the power 10 basis. Uh, when MMR is defective, the repetitive DNA regions are called microsatellites, and they become unstable and leads to the microsatellite instability or MSI. This instability dramatically increases the mutation rates in the genes and regulates the cell growth and apoptosis, predisposing the cells to uh, cancers of the colon and several other organs. And mutation in the key MMR genes disrupt the genomic integrity and allows the cancer promoting mutations to accumulate rapidly. So the third one is the base excision repair. What happens here is the base pairing of the 8-oxoguanine. Basically, it involves the oxidative damage to the guanine and produces the 8-oxoguanine, which is basically a highly mutagenic lesion, and can mispair with the adenine and cause the imbalance in the guanine cytosine ratio or the tyrosine adenine ratio. Uh, these specialized glycosylases such as the OGG1 and MYH-MTH1, they remove the 8-oxo-G and prevent these mutations from becoming fixed in the genome. And when this base excision repair components fail, the oxidative DNA damage can accumulate and increase the mutation rates. So basically, the failure of this base excision repair can lead to the this progression of the cancer. Now, the role of the telomeres and telomeres in genomic instability. So basically, what telomeres are and why they matter. So telomeres are the specialized DNA structure that are present at the ends of the chromosome and are made up of thousands of repeated TTAGG sequences. These repeats are not naked DNA. They are tightly associated with the telomere-specific proteins that form the protective cap. You can see here in this image. So this is the telomeric part, and their primary role is to distinguish the natural chromosome ends from the broken DNA, ensuring that the chromosome terminus is not mistaken for DNA damage. So telomerase basically maintains the length of the telomere. So this is the telomerase, and they contain the two essential components, which is basically the RNA template or the TERC, which provides the sequence for the telomerase synthesis, and the protein subunit, which is called the TERT, and this acts as the reverse transcriptase enzyme for the telomeres. So basically, this uses the RNA template and adds the TTAGG repeats to the chromosomal ends and compensates for the loss that occurs during the DNA replication. So it basically compensates the DNA damage losses which occurs during the replication. Now, why the telomere shortening is normal in somatic cells? So basically, in human somatic cells, telomerase is tightly downregulated, and as a result, the DNA polymerase cannot fully replicate the very ends of the lagging strand during the each cell division, and this is known as the end replication problem. As a result, the telomerase shortens progressively, and with every round of replication, it is shown that it keeps on shortening. So, studies shows that the human epithelial cells lose approximately 50 to 100 base pairs per population doubling, and as a result, a total lifetime of 2 to 4 kilobases. This gradual shortening acts as the biological clocks that limits the proliferation of the lifespan of the normal cells. The essential protective function of the telomeres, This can be listed as they prevent the chromatin ends from being recognized as a double-stranded breaks, and thereby activation of the DNA damage responses takes place. They protect the chromosome ends from the enzymatic downregulation by degradation by nucleases. They prevent end-to-end chromosome fusions, which would otherwise generate structurally abnormal chromosomes, and together these functions preserve the chromosomal integrity and genomic stability. So the telomere capping or the sheltering complex. So this is the sheltering complex, and it's a telomeric DNA is protected by multi-protein complex, which is known as the sheltering. So this duplex polymeric repeats bind to the TRF1 and the TRF2, so while a single-stranded overhang is bound by the pot1. These proteins interconnected by the defined other proteins and form a stable cap at the chromosomal ends, and this complex actively suppress the inappropriate activation of the non-homologous end joining or the NHEJ, and and as well as the homologous recombination or repair pathways at the telomeres. So telomere dysfunction activates the DNA damage response. So this dysfunction of this telomere can activate the response of the DNA damage. And when the telomeres become critically short or the sheltering complex are lost, the chromosome ends are exposed and perceived as the DNA breaks. And the DNA damage sensors such as the this complex of MRE, RAD50 and NBS1 uh, this complex detects the abnormality and activates the checkpoint kinases like the ATM, ATR, which results in the activation of the CHK1 and CHK2. And this signaling cascade culminates in activation of the P53, which triggers two powerful tumor suppressive responses like the cellular senescence and the permanent growth arrest or the apoptosis, and results in the prevention of the further divisions of the genetically damaged cells. So escape from these checkpoints and enters the crisis. So certain cells uh, uh, lose the p53 function and can escape the senescence despite the critical shortening of the telomeres. These cells continue to divide and enter the catastrophic phase known as the crisis, characterized by the rampant chromosomal instability. And during this crisis, the chromosomal ends fuse and break during mitosis and rejoin incorrectly and generate the non-reciprocal translational translocations or the NRTs, and they are extensively genomic rearrangements. This phase dramatically accelerates the mutations and accumulation. So the dysfunction in the telomeres promotes the oncogenic transformation. So in the image, you can see that the telomeres and the telomeres and cancer progressions are interrelated. So what telomeres are and why they matters is that the telomeres are the TTA double G GGG repeats and in the chromosomal ends, and they are the specialized DNA caps protecting the chromosome from chromosomal ends from degradation and fusion. So telomerase maintains the telomere length, and as a result, the ribonucleoprotein enzymes replenishes, and the telomere repeats in the stem or the germ cells takes place. Now the telomerase is downregulated, and the end replication problem causes the progressive shortening. This results in the essential protective functions like prevention of the DNA break recognition, protection from the nuclease degradation, and prevention of the end-to-end fusion. The telomere capping or the sheltering complex is the multi-protein complex which shields the telomeres from the DNA repair pathway. And the telomere dysfunction activates the DNA damage response like ATM, ATR kinases, and P53 activation, which results in the senescence or the arrest of the cell growth, as well as the apoptosis of the cell death. These are critically short telomere triggers the tumor suppressive responses. So, on the other hand, they can escape from the checkpoints and enter into the crisis, like certain cells which lose the p53 and bypass the senescence and leads to the rampant genomic instability. And as a result, short telomeres with p53 loss drive the selection of the pro-oncogenic genomes. They basically activate the oncogenes like the MYC and the RAS, and this alternative telomeric maintenance progresses the cancer progression, For example, like the cancer selected telomeres of the user ALT to bypass the short telomere barriers, which follows the telomere dysfunction, metastasis, and the malignancy. The metastasis, this metastasis can result into the spread of the cancers to the other distant organs, and they can also lead to the advanced malignancies. So another term associated with this is the aneuploidy, which is basically the chromosomal number errors, and this is basically arising from the like collective alignment and the equal segregation, and this results to the abnormal chromosomal number or the missing of the extra copies. So aneuploidy refers to the condition in which the cell has the abnormal number of the chromosomes, either missing or more chromosomes containing the extra copies. These abnormal karyotypes is a defining feature of the cancer cells and is thought to arise primarily due to the chromosomal instability. So this results into the mitotic errors, segregation errors, and cause the aneuploidy. Aneuploidy mainly results from the defects during the mitosis, especially improper attachment of the chromosomes to the mitotic spindle when the chromosomes do not segregate equally and the daughter cells inherit the wrong chromosome number. Normally, the spindle assembly checkpoint ensures that all the chromosomes are properly attached before the anaphase begins, and if this checkpoint fails, the cells proceed through the mitosis despite of the segregation errors. So meiotic aneuploidy occurs during the gamete formation and often leads to the spontaneous miscarriage. If the survival occurs, it results in the congenital disorders like the Down syndrome or the trisomy 21, as well as the Turner syndrome or XO. Uh, the mitotic aneuploidy arises in the somatic cells and is strongly associated with the advanced tumor, contributing to the tumor heterogeneity and the progression. Then comes the breakage fusion bridge or the BFB cycle, which is a driver of the chromosomal instability. What happens here is the telomeres at the end of a chromosome is lost or damaged, and as a result, the DNA replication of the chromosomes and the chromatids lack the telomeres at their ends of the replication. This basically leads to the absence of the telomeres and allows the sister chromatids to fuse, forming the dicentric chromosomes, and as a result the centromeres pull apart during the anaphase, creating a physical bridge. So the chromosomal breakage occurs, and as a result mechanical tension causes the chromosomes to break at the random location, which basically gives rise to the broken chromosomes, lacking the telomeres and repeating the cycle. So this broken chromosome still lacks the telomeres, and this BFB cycle repeats its subsequent divisions and genetic amplification, generation of the amplifications, deletions, and translocation until a telomere is restored via translocation or other chromosome. Another is the DNA damage signaling or the ATM-ATR checkpoint pathway, where the DNA damage is detected by the ATM and the ATR sensor kinase in normal conditions. But the activation of the checkpoint kinases, basically the checkpoints, the CHK1 and the CHK2, which results into the cell cycle arrest and the repair or the inhibition of the CDC25 cell cycle, allowing the DNA repair. But in P53 activation, the ATM and the ATR signaling stabilizes the P53, inducing the P21 and enforces the G1 and the G2 arrest. So apoptotic signals, they are the irreparable damage that triggers the P53 and ATM mediated pro-apoptotic pathways, and as a result, consequences of the checkpoint failure allows the DNA damage to divide, the causing the epigenetic instability, genomic instability, and the tumor growth. So senescence and immortalization has the role with RB and the P53 protein. So senescence is basically permanent arrest of the cell proliferation from which the cells rarely escape, and it's a powerful anti-tumor mechanism. But unlike the quiescent cells, the senescent cells do not resume divisions even when they are exposed to the growth factors like mitogenic signals. This triggers the senescence, and it's induced by the telomere shortening, oncogene activation, DNA damage, and oxidative stress. Senescent cells upregulate the cell cycle inhibitors such as the P16 and P14, which basically enforces the RB mediated cell cycle arrest as well as stabilizes the P53 inhibition and its degradation. The maintenance of the senescence is by the accumulation of these proteins and locks the cells to the senescent state, whereas the overexpression of these proteins alone is sufficient to induce the senescence. The loss of the RB or the P53 function allows the cells to escape the senescence by continuing the dividing and accumulate the mutations and ultimately achieve the cellular immortalization, which is a key step towards the cancer. Now, oncogenes overexpressed uh, are mutated or overexpressed uh, versions of the normal genes. So normal genes are called the proto and normal genes. When they convert into the oncogenes, they are called the proto-oncogenes, and they regulate the cell growth, cell division, and the cell survival, and acts like the stuck accelerator to promote the uncontrolled cell proliferation and are the hallmarks of the cancer. So proto-oncogenes in the normal cells contains the genes called the proto-oncogenes, which are essential for controlled cell growth and the division of the survival. But these genes encode the growth factors receptor signaling and proteins, which are transcription factors and cell cycle regulators. And under normal conditions, these proto-oncogenes are tightly regulated and ensures that the cells divide only when they're required and stop dividing when appropriate. The conversion of the proto-oncogenes into the oncogenes is when the proto-oncogenes undergo the dysregulation due to the mutation of the abnormal expression. And this change is usually caused by the gain of function and alterations, which means that the genes becomes the overactive or permanently switched on. And as a result, the cells receive the continuous growth signals or become the resistant to cell death, predisposing it to the cancer development. Activated oncogenes push the cells towards the uncontrolled proliferation and reduces the sensitivity to apoptosis, whereas the cells transformed by the oncogenes display the hallmark features of the malignancies. So these are the mechanisms of the activation of the oncogenes. One is the mutation or the point mutation or the indel mutations. The second is the overexpression, which is the gene amplification. And the third is the translocation of the chromosomal rearrangement. And translational outcomes also result into the activation of the oncogenes via the downregulated gene expression or the gene fusion, where the fusion gene and the chimeric protein enhances the activity. And certain examples of the oncogenes are the CMYC gene, which is a very strong in immunoglobulin enhancer and continuous like, and has a characteristics of continuous expression. So the consequences of the gene amplification are like the double minute chromosomes or the heterogeneous staining regions or the HSR, which is basically extremely overexpressed growth advantage. And an example is the CML, which is basically the BCR and the ABR fusion. So ABL gene and the BCR gene, they fuse to form the BCR ABL fusion gene, and they constitute tyrosine kinase activity, which basically gives rise to the uncontrolled proliferation. This is followed by the viral contribution, and where the RNA via the reverse transcription forms the DNA and uh, integrates and activates the cellular oncogenes and disrupts the regulation. So this is a figure of the sequential activation of the oncogenes in the cell cycle, cell signaling, and the cancer. So basically, there are the growth factors which initiates the signals, and they basically follow an autocrine loop, and they are self-sufficient in the cancer cells. Cancer cells produce their own growth factors for the unregulated proliferation. This is followed by the growth factor receptors, or the RTKs, receptor tyrosine kinases. Certain mutated receptor tyrosine kinases, like the EGFR or HER2, like in breast cancer, they are ligand dependent, independent activation, and their via mutation overexpression drives continuous signaling. Then comes the adapter proteins, which links the signalings. So basically, GRB2 bridges the receptors to signal the enzymes like SOS1. So the guanine nucleotide exchange factors, or the GEFs, like the SOS1, activates the small GTPases, amplifying the signals. There are the membrane-associated G proteins, also the RAS hub. These are the mutations that logs the RAS in permanently active state and continuously signaling for the proliferation or the survival. Then there comes the RAF or MAP kinase, which are the serine threonine protein kinases, like AMPK pathway kinases, and this initiates the MAP kinase cascade, leading to the nuclear responses and BRAF mutations and cause the constitutive activity. The certain nuclear oncogenes are the transcription factors, for example, the BCR-ABL fusion kinases, and the conversion of the cytoplasmic signals into the long-term gene expression changes the regulating growth and the survival. The cytoplasmic survival oncogenes sustain the malignancies, like the AKT pathway inhibits the apoptosis and promotes the metabolism, ensuring the cancer cell survival. The growth factors, they are the initiating the signals. So they are the initiating signals in the oncogenes. Uh, they initiate the signals in the oncogenes, and in normal physiology, they are the paracrine or the endocrine signals. Whereas in cancer physiology, they are the autocrine simulations. In normal cells, the cells basically they target the neighboring cells. The growth factors from one cells will target the neighboring cells, whereas in the cancer cells, the growth factors from the same cells will target the receptors of the same cell. And this is self-sufficiency. So cancer cells can produce their own growth factors and receptors, leading to the unregulated proliferation, the independent external signals, and independently of external signals. So the growth factor receptors or the RTKs, receptor tyrosine kinases, they're the signaling and the dysregulation in the cancer. So in normal signaling, the ligand dependent activation occurs, whereas in the cancer, the ligand independent activation occurs. So there is the mutation and the overexpression of the these receptor tyrosine kinases, and they give the uncontrolled signaling, and as a result, uncontrolled cell division occurs and tumor growth can occur. Then comes the adapter proteins in signaling, which has the GRB2 as a molecular bridge, and oncogenic signaling, which is basically amplified form, whereas in normal cells it's in controlled form. So these activated RTKs have the controlled signaling for the protein signaling like GRB2 molecules, whereas in oncogenic signaling they are amplified, and you can see there can be multiple of signaling or the overexpression, and this is expressed, for example, in GRB2 and SOS in primary mechanism in the bladder cancer. So to summarize this, the overexpression of the adapter proteins like the GRB2 amplifies the signaling without receptor mutations and drives the oncogenesis. So the GEF, or the guanine nucleotide exchange factors, and they are the they activate the molecular switches. So they are like SOS1 and activates the small GTPase by catalyzing the GTP exchange, or the ratio of the exchange of the guanosine diphosphate and the guanosine triphosphate exchange. And the dysregulation leads to the persistent downstream signaling, which is a common feature in cancer. So it is an emerging therapeutic target and is being studied globally. Now there are the membrane-associated G proteins, where RAS is a central oncogenic hub. In normal RAS cycle, it's tightly regulated switch, whereas in oncogenic RAS, it's a permanently activated state. So oncogenic mutations prevent the GTP hydrolysis, resulting the locking of the RAS in permanently active state that continuously drives the cancer cell signaling. Now comes the serine threonine protein kinases, or the RAF and MAP kinase signal. In normal cells, the RAS dependent activation and the cascade is regulated, whereas in the cancer cell, the BRAF and the V600 mutation causes the constitutive RAS independent activity, which drives the unregulated growth. So, certain therapies block the mutated BRAF, and the BRAF V100 V600 E mutation leads to the constitutive MAP kinase pathway activation, which basically so targeted therapies like the vemurafenib inhibit the mutated kinases. Now, cytoplasmic tyrosine kinase in cancer, and these are the different types like SRC, JAK, and the BCR-ABL. Like dysregulated cytoplasmic tyrosine kinase act as the powerful oncogenic drivers and are the key therapeutic targets in various cancers. The transcription factors in the cancer signaling are AP1, then STAT proteins, then CMYC, and NFKB. So the transcription factors such as the AP1, STATs, and all these, they convert the cytoplasmic signals into the long-term gene expression changes. For example, the AP1 or the FOSGen regulates the proliferation and the metastasis, the downstream of the growth factor signaling. STAT protein activates the JAKs, activated by JAKs and Janus kinase. So basically, it translocates to the nucleus and induces the genes involved in the survival angiogenesis. The CMYC is a master regulator of the growth and the metabolism. And in Burkitt lymphoma, the chromosomal translocation places the MYC under the immunoglobulin enhances, causing the uncontrolled proliferation. The MYC dysregulation occurs in over 70% of the human cancers. The NFKB promotes the cell survival and inflammation, and its chronic activation supports the tumor growth and resistances to apoptosis, making it important therapeutic target. The cytoplasmic survival of the oncogenes by the PI3K and AKT pathway. The oncogene signaling ultimately covers on the cell survival pathways like the PI3K, AKT, and which inhibits the apoptosis and promotes the metabolism and protein synthesis. AKT phosphorylation on the pro-apoptotic proteins such as the BAD ensures the survival is even under stress. Uh, these pathways enable the cancer cells to withstand the genomic instability and hostile the microenvironment. So there are the different classes of the oncogenes. These are the growth factors, then the growth factor receptor kinases or the RTKs. There are the membrane associated G proteins, which are the RAS and RAS-GTP, and locked in the GTP bound state. Then there are the serine threonine kinase proteins like MAP kinase pathway, as well as the which basically after mutation causes the uh, cancers. And there is the cytoplasmic tyrosine kinases. There is the nuclear protein transcription factors and cytoplasmic proteins of the cell survival. Now, the growth factors can act as the oncogenes, and the cell growth and the proliferation are subject to the regulation by external signals that are typically transmitted to the cell in the form of the growth factors that bind to and activate the specific growth receptors. So basically, the growth factors are the results in the gene amplification, and for example the ERBB2, and this results in the gene amplification or mutation, for example ERBB2 and EGFR, and this results in the signaling molecule activation and activation of the protein kinase cascade, and as a result the transcription factors and the DNA repair complex that acting on the target gene, this transcription factors amplification or the translocation can take place, and for example in the MYC and the ERG, and as a result the target gene activation takes place, and the target gene activation in the cancer can lead to the cancer. For example, the activation of the cell cycle regulatory gene like cyclin D can lead to the activation of cancer. So defined chemical signaling forms can be autocrine, which are the self targets self cells, which are the cells that targets itself, and then there can be the signaling across the gap junctions, where a cell targets a cell connected by a gap junction. Then there can be paracrine growth factors, where a cell targets a nearby cell. And then there can be endocrine, where a cell targets a distant cell through the bloodstream. Now, growth factors receptors are typically the receptor tyrosine kinase, which are present in the plasma membrane. And upon binding with the ligand, these receptors dimerize and activate intracellular kinase domains, which initiate the signaling cascades. So some of the mutations, overexpressions, or constitutive activation of the receptors allow the cells to signal continuously, even in the absence of the ligand, and driving the uncontrolled proliferation. So this class includes some of the most important therapeutic targets in the cancer. And the growth factor receptors are composed of the three domains. These are the extracellular domain or the ligand binding domain. Then comes the hydrophobic transmembrane domain, and there is the cytoplasmic or the kinase domain. Again, there is the phosphorylate tyrosine residue, which is there in the another domain. So the receptors are also called the receptor tyrosine kinase, essential to the intracellular signaling. Due to the mutation in the domain, the signaling promotes unregulated cell proliferation, and autophosphorylation can result into the signaling cascades, which further regulates the proliferation. So these three domains basically they constitute actively the receptor tyrosine kinase of the growth factor receptors. And one of the example are the EGFR, which undergoes a transition from the inactive monomeric form to an active homodimer, and some other examples are listed here. So ERBB2 has no known direct activating ligand and may be an activated state constitutively or become active upon heterodimerization with other family members like EGFR. EGFR dimerization stimulates its intrinsic intracellular protein tyrosine kinase activity. And as a result of the autophosphorylation of several tyrosine, which are basically the Y residues in the C terminal domain of the EGFR takes place. And these includes the Y992, Y1045, and many others. Now there are GEFs, or the guanine nucleotide exchange factors. So basically these are the proteins or the protein domains which are involved in the activation of the small GTPase, and they act as the molecular switches in intercellular signaling pathways and have many downstream targets. The most well-known GTPase comprises the RAS superfamily and are involved in the essential cell processes such as the cell differentiation, proliferation, cytoskeletal organization, as well as the vesicle trafficking and the nuclear transport. GTPase are active when bound to the GTP and are inactive when bound to GDP. This allows their activity to be regulated by the GEFs, and that is the guanine and nucleotide exchange factors, and the opposing GTPase activating proteins, or the GAPS. GDP dissociates from the inactive GTPases very slowly. The binding of the GFs to the GTPase substrates catalyze the dissociation of the GDP, allowing a GTP molecule to bind in its place. So basically, in the image, you can see like the inactive GTPase bound. It promotes the dissociation of the GTP when the GTP is released, and the active GTPase sorry when the GTP comes and binds to the GEFs, uh, or the GTPase, they activate the GTPase, and basically with binding of the GTP, and this results into the different downstream targets like cell differentiation, proliferation, cytoskeletal organization, as well as vesicle trafficking and nuclear transport. Now, talking about the G proteins, the signaling that is initiated by the growth factors activating their receptors passes next to the membrane-associated guanine nucleotide binding proteins, and these are called the G proteins. The prototypical member of this family of these oncogenes is the RAS, or the rat sarcoma oncogene. There are three RAS genes like HRAS, KRAS, and NRAS, which are basically the neuroblastoma, NRAS, and these genes differ in their expression patterns in different tissues, and they have been found to have point mutation in the cancers, including the liver, colon, KRAS in the colon, and HRAS in bladder. and they are also found in the pancreas and lung cancer. And this leads to the constitutive signaling of the genes involved in the proliferation. uh, RAS is post-transcriptionally modified by attachment of the farnesyl fatty acid moiety to the C terminus and is essential for activity of the RAS oncogene. The constitutively active RAS is the oncogene mutation and are concerned within the two hotspots, which is around the codons of 12 and 61. RAS is permanently in an on state when the constitutively active RAS is, which is one of the most, which contains mutations that prevents the GTP hydrolysis, and thus it logs the RAS in permanent on state. The most common mutations that are found in the residues of G12 in the P loop and the catalytic residues is Q61, and the glycine to valine mutation at the residue of the 12, it renders the GTPase domain of the RAS, which is insensitive to inactivation by gap and thus stuck in the on state. Several other G proteins like RAS binds to both GDP and GTP, but active only when bound to GTP. uh, NF1 or the neurofibromin 1 is a gene, is a gap in the chromosome 17, which is a negative regulator of RAS and activates the P21 activating protein. So mutated, when NF1 is mutated, it gives rise to the neurofibromatosis, and it's a common symptom of NF1, which includes the scoliosis, and which is basically the curvature of the spine and leaning disabilities, and the vision disorders and epilepsy. So there are certain RAF serine threonine protein kinase. The RAF serine threonine protein kinase is also known as the proto-oncogenes, and they are the protein kinases like EC 2.7.11.1, and is a kinase enzyme that is that phosphorylates the OH group of the serine and the threonine. These are the virus-induced and rapidly accelerated fibrosarcoma. So RAF then initiates a cascade of the mitogen activated protein kinase or MAP kinase, which culminates the nucleus with activation of the genes containing the ELK1 and transcription factor, that transcription factor binding sites. Another group is the phosphate inositol 3 kinase, which initiates the signaling pathway for cell survival. The PI3K phosphorylates the phosphatidylinositol and triphosphate, which is an important intracellular second messenger. The PI3K consists of a catalytic subunit, which is called the P110, and a regulatory subunit, the P85. And the PI3K phosphorylates the protein kinase B, or which constitutively is also the AKT-PKB, on the serine and the threonine residues, which in turn modulates the cellular processes like glycolysis and translation and initiates an elongation. So RAF1 was reported to directly phosphorylate the and inactivate the RB protein, which leads to the cell cycle progression. So this slide focuses on the BRAF mutations and the role in the cancer, particularly through the MAP kinase signaling pathway. Under normal conditions, the BRAF functions as a serine threonine kinase downstream of RAS in the MAP kinase pathway. It becomes activated only when the RAS is in its GTP bound state, and once activated, the BRAF phosphorylates the MEK, which in turn activates the ERK signaling, leading to the controlled processes such as the cell proliferation and the differentiation. This ensures that the cell growth occurs only in the response to the proper external signals. In cancer mutations in the BRAF gene, which are common in the BRAF gene, occurring in approximately 20% of the human cancers. These mutations are especially significant in the cancers such as the melanoma, thyroid cancer, colorectal cancer, and some lung cancers, making BRAF one of the key oncogenic drivers in the solid tumors. The most important mutation is the V600E mutation. Uh, the V600E, where the valine at the position 600 is replaced by the glutamic acid. This substitution mimics the phosphorylation within the activation loop of the protein, forcing BRAF into the permanently active state, and as a result, the kinase no longer depends on the upstream RAS signaling. Because of this mutation, the BRAF constitutively becomes active and continuously sends the signals to the MAP kinase pathway, whereas the MEK and the ERK remains persistently active, leading to uncontrolled cell proliferation and increased survival and tumor progression. This happens even in the absence of the growth signals. Clinically, this mutation can be detected using the immunohistochemistry, which allows the identification of the tumors driven by BRAF V600E. And this is important because it directly influences the treatment decisions, especially the use of the targeted therapies. The two major therapeutic approaches are highlighted here. One involves the sorafenib, a multikinase inhibitor that targets the BRAF in its inactive conformation while preventing the downstream signaling. It is used in the cancer such as the liver, kidney cancer. And the second approach is the this drug called vemurafenib, which is a highly sensitive inhibitor that specifically targets the active form of the BRAF V600E. It binds the ATP binding site of the kinase and is particularly effective in treating the advanced melanoma patients with this mutation. Overall, this slide demonstrates how the single point mutation of the BRAF can shift a tightly regulated signaling pathway into a continuously active oncogene driver, and how the understanding this mechanism has enabled the development of the targeted therapies. This slide explains the structure and function and the oncogenic role of the SRC family, non-receptor tyrosine kinase. The SRC family consists of the 9 members, including the CSRC, YES, FYN, FGR, YRK, LYN, BLK, HCK, and LCK. These proteins are not uniformly expressed across all tissues. Rather, SRC, YES, and FYN are found in most cell types, whereas other members are predominantly expressed in the hematopoietic cells, indicating their specialized roles in immune and blood cell signaling. A key feature of the SRC protein is their post-transcriptional modifications, and they undergo the myristoylation at the end terminus, which allows them to associate with the inner surface of the plasma membrane. This localization is crucial because many signaling events occur at the membranes where the receptors and the adapted proteins are present. Structurally, SRC protein contains 3 important domains, which is known as the SH1, SH2, and the SH3. Uh, the SH1 domain is the catalytic domain, which is responsible for the tyrosine kinase activity. It uses the ATP to phosphorylate the target proteins on the tyrosine residues and thereby propagating the intracellular signaling. The SH2 and the SH3 domain are regulatory domain that mediate the protein-protein interactions. The SH2 domain specifically binds to the phosphorylated tyrosine residues on other proteins, while the SH3 domain interacts with the proline-rich regions. These interactions allow SRC to be recruited into the signaling complexes and help regulate its activity. In its inactive state, the SRC adopts a close conformation, and this is maintained through the intramolecular interactions involving the SH2 and the SH3 domain, which bind the internally and suppress the kinase activity. This serves as the form of the negative regulation and preventing the unnecessary and excessive signaling. Acha. When these regulatory interactions are disrupted, either by the mutations in the SH2 or SH3 domains, or by dephosphorylation of the inhibitory sites, the SRC shifts into an open and active conformation. In this state, the kinase becomes constitutively active and continuously phosphorylates the downstream targets. This uncontrolled activation leads to the persistent signaling pathways that promotes the cell proliferation, survival, and the migration, all of which can be contributed to oncogenic transformation. Because of this, the SRC kinase are important targets in the cancer cell therapy, and efforts are going on to develop the drugs that inhibits the abnormal signaling activity. Here we see how the SRC kinase is activated and its role in cancer and how it is targeted therapeutically. The activation of the SRC occurs when Y527 is dephosphorylated. This disruption this disrupts the internal bonding between the SH2 domain and the phosphorylated tail, causing the structure to open up. As the protein unfolds, the SH2 and the SH3 domain disengages, exposing the kinase domain. The SRC then undergoes the autophosphorylation at another site, which is the Y416, which fully activates its kinase activity and allows it to phosphorylate the downstream targets. Once activated, the SRC integrates the signals from the multiple upstream receptors, and these includes the addition receptors, receptor tyrosine kinase, G protein coupled receptors, and cytokine receptors. Through these inputs, the SRC acts as a central signaling hub that connects the extracellular signals to intracellular responses. The activated SRC promotes several processes that are critical for cancer progression. These includes the increased cell survival, enhanced angiogenesis, uncontrolled proliferation, and the ability of the cells to invade the surrounding tissues. In many cancers, the SRC becomes constitutively active due to the mutations, increased expressions, or abnormal upstream signaling, leading to persistent oncogenic signaling. This this dysregulation has been observed in a significant proportion of the tumors, including the cancers of the colon, liver, lung, breast, and pancreatic cancers. Because of its central role in cancer signaling, SRC has become an important therapeutic target, and one of the key drugs used to inhibit the SRC activity is dasatinib, which is a tyrosine kinase inhibitor. Dasatinib binds the kinase domain and blocks its activity, thereby preventing the downstream signaling. It is currently approved for the treatment of the chronic myeloid leukemia and Philadelphia and chromosome positive acute lymphoblastic leukemia. It is also being explored in the clinical trials of other cancers such as the breast cancer, prostate cancer, and many others. Now, talking about the BCR-ABL fusion oncogene, which is a central to certain leukemias. The process begins with a chromosomal abnormality known as the reciprocal translocation between the chromosome 9 and the chromosome 22. In this event, a segment of the ABL gene from the chromosome 9 is transferred to chromosome 22, where it fuses with BCR gene. This rearranged chromosome is called the Philadelphia chromosome, and its presence is a key diagnostic feature of the chronic myelogenous leukemia. As a result of this translocation, a hybrid gene known as the BCR-ABL is formed, and this gene encodes a chimeric protein that combines the functional element from both BCR and ABL. The BCR portion contributes regulatory and protein interaction features, while the ABL portion contains the tyrosine kinase domain. While under normal conditions, ABL kinase activity is tightly regulated. However, in the BCR-ABL fusion protein, this regulation is lost. The kinase becomes constitutively active, meaning it is always on regardless of external signals. This persistent activation is a key driver of the oncogenic signals. The fusion protein also interacts with the adapter protein such as the GRB2 through its SH2 domain, and this interaction helps to recruit the downstream signaling molecule and activates the pathways like RAS signaling and cascade. These pathways transmit the continuous signaling growth signals and survival signals to the cell. As a consequence, the cells overexpress the BCR-ABL and show several abnormal behaviors. They become less dependent on the external growth factors, which means they can proliferate without normal regulatory signals. Their adhesion properties are altered, and they allow them to detach more easily from their normal environment, which most importantly, they exhibit enhanced survival and rapid proliferation and contributes to the uncontrolled expansion of the leukemic cells. Overall, this slide demonstrates how the single chromosome translocation can generate a fusion protein with constitutive kinase domain activity, leading to continuous signaling, loss of the growth, and development of the cancer. Now talking about the transcription factors which can act as the oncogenes. This slide focuses on AP1, also known as the activated protein 1. The AP1 is the nuclear transcription factor that regulates the gene expression in response to various signals. It belongs to a broader group of the transcription regulators that includes the proteins like CMYC, STAT, and NFKB, all of which play important role in the controlling the cell growth. The structurally AP1 is not single protein but a complex formed by dimerization of the members of two protein families, the JUN family and the FOS family. These proteins interact through a specialized region known as the leucine zippers, which allows them to bind to each other and form the stable dimers. The most active and the functional significant form of the AP1 is the heterodimer composed of the FOS and the JUN. The JUN-JUN homodimers are less active, whereas the FOS-FOS homodimers are rarely formed. Once formed, the AP1 complex binds to the specific DNA sequences known as the response elements, such as the TPA response element, through which this binding AP1 regulates the transcription of the genes involved in the key cellular processes. These includes the promotion of the cell proliferation, regulation of the differentiation, and control of the apoptosis. Because these processes are central to maintain and normal cell behavior, the dysregulation of the AP1 activity can contribute to the oncogenes. The activity of the AP1 is tightly controlled by the external signals, particularly those originating from the growth factor receptors like EGFR. When such receptors are activated, they trigger intracellular kinase cascades that ultimately lead to the activation of the AP1 in the nucleus. This connection illustrates how the signals from outside the cell can directly influence the gene expression programs inside the nucleus. In cancer, this regulatory system becomes disrupted, and overactivation of these signaling pathways or increased expression of these AP1 components can lead to the persistent transcription of the genes that promote the proliferation of the survival while suppressing the normal apoptotic responses. This slide explains the STAT protein family and how the signal transduction leads to the activation of the transcription through the JAK and the STAT pathway. So the STAT proteins are intracellular transcription factors that play essential roles in regulating the immunity and cell proliferation, apoptosis, and differentiation. In mammals, there are the 7 members of this family, including the STAT 1, 2, 3, 4, 5, and 6. 5 has 5A and 5B, and these proteins remain inactive in the cytoplasm until they are activated by the upstream signaling events. Structurally, the STAT protein contains the several conserved domains. At the N terminus, there is the domain involved in the dimerization, which is followed by the coil domain that helps in the protein interactions and nuclear localization. The DNA binding domain allows the STAT protein to attach to the specific gene promoters, where a linker domain connects to the SH2 and SH2 domain, which is critical for the recognition of the phosphorylated tyrosine residues and enabling the dimer formation. Finally, the C-terminal domain functions as the transcriptional activation region. The signaling process begins when a cytokine or the growth factor binds to the receptor on the cell surface, and this activates the associated Janus kinase or the JAKs, which phosphorylates the specific tyrosine residues on the receptor. The STAT proteins are then recruited to these phosphorylated sites and through their SH2 domain and become phosphorylated themselves by the JAKs. Once phosphorylated, the STAT protein dissociates from the receptor and they form the dimers through the interactions between their SH2 domain and the phosphotyrosine residues. The dimerization is essential for their activation. The STAT dimers then translocate into the nucleus through the nuclear pore complex with the help of the importin proteins. Inside the nucleus, the STAT dimers binds to the specific DNA sequence, which is also known as the gamma activated sites in the promoters of the target genes. This leads to the transcription of the genes involved in the cell growth, survival, immune response, and the differentiation. After completing their function, the STAT proteins are dephosphorylated by the nuclear phosphatase, which leads to their inactivation, and they are then exported back to the cytoplasm. And they are ready for the another cell cycle of activation and the cycle of activation. In the cancer, this pathway is often dysregulated. Persistent activation of the STAT proteins, especially the STAT3 and the STAT5, leads to the continuous transcription of the genes that promote the tumor growth, angiogenesis, and enhanced survival and immune evasion. To continue with this, we can look at the overview of the JAK-STAT signaling pathway, covering the structural mechanism and nuclear transport and the clinical relevance. Starting with the structure, JAK proteins, which include the JAK1, 2, 3, and TYK2, are associated with the cytoplasmic portions of the cytokine receptors. Each JAK protein contains multiple domains, including the firm domain for the membrane association, an SH2 domain, a pseudokinase domain that regulates the activity, and a kinase domain that responds to the catalytic function. STAT proteins, on the other hand, contain the SH2 domain for the dimerization and receptor binding, the DNA binding domains for the gene regulation, and nuclear localization signals that allow them to enter the nucleus. The signaling mechanism begins when a cytokine binds to the receptor, causing the receptor dimerization. This brings two JAK molecules into the close proximity, allowing them to phosphorylate each other in a process called the transphosphorylation. This activation increases their kinase activity, and the activated JAKs then phosphorylate the specific tyrosine residues on the receptors, creating a docking site for the STAT proteins. The STAT proteins are recruited to the receptor via the SH2 domains and the subsequently phosphorylated by the JAKs. And once phosphorylated, the STAT proteins dissociate from the receptors and form the dimers, and these dimers represent the active form of the STAT and are capable of transmitting uh, the signals further into the cells. The next step involves the nuclear transport, where the STAT dimers binds to the importing proteins and pass through the nuclear pore complex into the nucleus. Once inside, they are released and bind to specific DNA sequences, initiating the transcription of the genes involved in the proliferation, also in survival and immune response. Clinically, dysregulation of this pathway is associated with several cancers. For example, high STAT3 activity is observed in melanoma, prostate cancer, and the breast cancer, where it drives the expression of the genes such as the BCL2 and the CMYC. In leukemias and lymphomas, the mutation in the JAK2, particularly in specific exons, lead to the constitutive activation of the pathway. Because of its importance in the disease, this pathway is a major therapeutic target where JAK inhibitors such as the ruxolitinib and tofacitinib are used to block the abnormal signaling. Additionally, efforts are being made to develop the STAT inhibitors, especially the targeting the STAT3 and to reduce the tumor growth, which results in improved patient outcomes. This slide explains the CMYC oncogene and its function, mechanism of the dysregulation in cancer, and the challenges associated with the targeting it therapeutically. The first section focuses on the Burkitt's lymphoma and the chromosomal translocation. Normally, the CMYC gene is located in the chromosome 8, and in Burkitt's lymphoma, a reciprocal translocation occurs between the chromosome 8 and the chromosome 14. This places the CMYC gene under the control of the immunoglobulin heavy chain enhancer, which is highly active in the B lymphocytes. As a result, the CMYC becomes overexpressed, leading to uncontrolled cell proliferation and tumor development. The second section describes the regulation through the APC and the beta-catenin pathway. Under normal conditions, the APC complex acts as a negative regulator by promoting the degradation of the beta-catenin. However, when APC is mutated or the beta-catenin is stabilized, the beta-catenin accumulates and translocates to the nucleus. There, it interacts with the transcription factors like TCF4 to activate the CMYC transcription. This leads to the increased expression of the genes involved in the cell cycle progression, protein synthesis, stress response, and ultimately driving the hyper proliferation. The second section describes the regulation through the APC and the beta-catenin pathway. Under normal conditions, the APC complex acts as the negative regulator by promoting the degradation of the beta-catenin. Similarly, the third section highlights the oncogenic action of the CMYC at the protein level. The CMYC is initially an unstructured protein, but it becomes the functionally active when it dimerizes with another protein called the MAX. The interaction occurs through a helix loop leucine zipper domain. The MYC MAX complex binds to the specific DNA sequence known as the E-box elements and activates the transcription of the genes that promotes the cell growth, metabolism, and division. The dysregulation of the mechanisms is observed in a large proportion of the cancer, including the breast, liver, colorectal, and prostate cancer. The final section addresses why CMYC is considered difficult to target therapeutically. Unlike many enzymes, CMYC doesn't have a well-defined active site or binding pocket, similar for the small molecule drugs. It is localized in the nucleus, making it inaccessible to certain therapeutic agents like antibodies. Addition to its structure, the highly dynamic and the disordered, which complicates the drug design. The current strategies focuses on disrupting the interaction between the MYC and MAX or targeting the regulatory regions involved in its function. The following slide explains the NF-kB signaling pathway and its role in cellular functions, as well as its importance in the cancer and inflammation. NF-kB is a protein complex that plays a central role in regulating the immune response, cytokine production, and cell survival. Under normal conditions, NF-kB or nuclear factor kappa B exists in the active state in the cytoplasm. It is bound to an inhibitory protein called IKB, which prevents it from entering the nucleus and activating the gene transcription. So IKB is the inhibitor of KB, and these signals activate the IKK complex, which phosphorylates the IKB. This phosphorylation marks the IKB for degradation of the proteosomes, and once the IKB is degraded, NFKB is released from the inhibition. The active NFKB complex, typically composed of the P65 and the P50 subunits, then translocates into the nucleus. Inside the nucleus, it binds into the specific DNA sequence known as the KB sites and initiates the transcription of the genes involved in the inflammation, the immune response, the self-proliferation, and survival. The structure of the NFKB proteins includes the conserved domain such as the REL, which is the homology domain, which is responsible for dimerization and DNA binding and nuclear localization. Some subunits also contain activation domains that enhances the gene expression, while others can act as the transcriptional repressors depending on their composition. In cancer, the chronic inflammatory conditions like NFKB signaling pathway becomes persistently activated. This leads to the continuous expression of the genes that promotes the cell survival, inhibits the apoptosis, and support the chronic inflammation, all of which contributes to the tumor development and the progression. Because of its central role in diseases, NFKB is important therapeutic target, and several strategies have been developed to inhibit this pathway. For example, the proteasome inhibition, upstream signaling molecule, as well as directly inhibiting the NFKB subunits. Overall, this slide highlights how NFKB acts as a regulatory enzyme, linking the external stress signal to gene expression, and how its dysregulation contributes to the cancer and inflammation. The tumor suppressor genes are also known as the anti-oncogenes and act as the guardians of the cell. The tumor suppressor genes are essential for protecting the cells from the cancer by regulating the cell cycle, promoting the apoptosis, and maintaining the genomic stability. Unlike oncogenes, which drive the cell growth, tumor suppressor genes act as brakes that prevent the uncontrolled proliferations. When these genes lose their function, cells can grow uncontrollably and progress towards cancer. One of the primary functions is the repression of the cell cycle genes. The tumor suppressors inhibit the expression of the genes required for the cell cycle progression, effectively halting the cycle when conditions are not suitable for the division. This ensures that the cells do not divide inappropriately. Another critical role is the coupling of the cell cycle to DNA damage responses. When the DNA damage is detected, the tumor suppressor pathways decide whether the cells should pause and repair the damage or undergo apoptosis. If the damage is too severe, this prevents the propagation of the mutation of the daughter cells. The tumor suppressor genes are also directly induce the apoptosis. They activate the programmed cell death mechanisms to eliminate the damaged or abnormal cells and thereby protecting the organisms from the potentially carcinogenous transformations. In addition, they play a role in suppressing the metastasis and maintaining the cell adhesion by preserving the contact inhibition and preventing the cell detachment. They help ensure that the cells remain in their proper location and do not spread to other tissues. The tumor suppressor genes can be classified into three functional categories. The gatekeepers directly control the cell proliferation and apoptosis. They act as the first line of defense. The caretakers maintain the genomic integrity by regulating the DNA repair mechanisms and cell cycle checkpoints, whereas the landscapers influence the cellular environment, ensuring that the surrounding conditions do not favor the abnormal cell growth. The examples include the APC, which is the functions as the gatekeeper, and MSH2, which acts as the caretaker involved in the DNA mismatch repair. The tumor suppressor genes through the two-hit hypothesis. It highlights the role of the RB protein in the cell cycle regulation. The two-hit hypothesis proposed by the Cudson describes how tumor suppressor genes must typically lose both the functional copies of the copies to contribute to cancer development. In normal cells, both alleles of the tumor suppressor gene, such as the RB, are intact and produce the functional proteins, allowing the proper control of the cell growth. In the first step, one allele becomes mutated. The cell is now heterozygous, but the remaining normal allele is still sufficient to maintain the function. At this stage, there is usually no disease because the tumor suppressor genes follow a recessive pattern at the cellular level. In the second step, the remaining normal allele is also lost or mutated. This results in the complete loss of the function, leading to the uncontrolled cell proliferation and tumor formation. This explains why both the copies of the tumor suppressor genes are generally required to be inactivated for the cancer development. However, there are exceptions to this rule. In some cases, such dominant negative mutations in the p53, a single mutated protein, can interfere with functions of the normal protein, whereas in other situations, such as the haplosufficiency, the loss of the just one allele may be enough to disrupt the normal function, and this can be seen in the genes like p27 or NF1. The slide then focuses on the retinoblastoma proteins, or RB, which is a key tumor suppressor involved in the controlling of the G1 to the S phase, the translation of the cell cycle, and the RB gene is located on the chromosome 13. This mutation in this gene are associated with the retinoblastoma, a childhood eye cancer, as well as other cancers such as the breast and the lung and the bladder cancer. This functionally RB regulates the activity of the transcription factor called the E2F in its active hypophosphorylated state. The RB binds to the E2F and prevents it from the activating the genes required for DNA synthesis. This effectively blocks the progression of the G1 phase to the S phase, halting the cell division. When the cells receive appropriate growth signals, the cyclin-dependent kinases phosphorylates the RB protein, and this phosphorylation inactivates the RB, causing it to release the E2F. Once the E2F activates the transcription of the genes necessary for the DNA replication, allowing the cell cycle to proceed. So this slide explains how the APC tumor suppressor regulates the WNT/beta-catenin pathway and how its disruptions leads to the cancer. In normal off state, beta-catenin state, the beta-catenin is phosphorylated and continuously degraded. This is achieved through a destruction complex composed of the APC, exin, and GSK3, where beta-catenin is phosphorylated and is targeted for the proteasomal degradation. And as a result, the beta-catenin cannot enter the nucleus, and the gene expression related to the proliferation remains repressed. When the WNT signaling is activated, the destruction complex is inhibited. So the beta-catenin becomes stabilized and that accumulates in the cytoplasm and eventually translocates into the nucleus. There it binds to the TCF and LEF transcription factors and activates the genes such as the CMYC and the cyclin D1 and promotes the cell proliferation. In cancer mutations, the mutations in the APC or the beta-catenin disrupts this regulation, and the APC loss or the beta-catenin mutations prevents the degradation. This leads to the continuous beta-catenin accumulation, and as a result, in constant activation of the proliferation of the genes, the driving uncontrolled cell division and tumor development occurs. And this happens especially in the colorectal cancers and the familial adenomatous polyposis. The TGF beta, which is a double-edged sword in cancer, it highlights the dual role of the TGF beta in cancer, acting as both tumor suppressor and the tumor promoter, and it depends on the stage and the cellular context. In normal epithelial cells, the TGF beta inhibits the growth by inducing the cell cycle arrest, and it activates the SMAD protein, which increases the expression of the CDK inhibitors such as the P15 and the P21, while suppressing the CMYC and prevent the phosphorylation of the RB protein and blocks the progression of the G1-2S phase. However, in other contexts, such as the endothelial cells, the TGF beta can stimulate the growth. In early cancer stages, its primary role is tumor suppression through inhibition of the proliferation, while in advanced cancer stages, the role reverses. The tumor cells exploit the TGF beta signaling to promote the invasion and metastasis and angiogenesis through the VEGF, and this immune evasion suppresses the immune responses. Additionally, the mutations or the loss of the TGF beta receptors impairs its suppressive function, and these alterations are observed in the cancer like colorectal and the breast and the oral cancers. This allows the tumor cells to escape the growth inhibition while retaining the pro-tumorigenic effects. The P53 is a tumor suppressor and also the guardian of the genome, and it explains its central role of P53 in maintaining the genomic stability. P53 is activated in response to the cellular stress and DNA damage through the signaling pathways involved in the kinases such as the ATM, ATR, and MAP kinases. Once activated, the P53 functions as the transcription factor that regulates the genes involved in the cell cycle control, DNA repair, and apoptosis. Structurally, P53 contains the domains responsible for DNA binding, transcriptional activation, tetramer formation, and damage recognition. Upon activation, the P53 induces the cell cycle arrest at the G1 or S checkpoints and allows the time for the DNA repair by activating the repair proteins. If the damage is repairable, the P53 triggers the apoptosis to eliminate the damaged cells, and it also plays a role in preventing the angiogenesis while maintaining the genomic stability by limiting the mutation accumulation. Through these coordinated responses, the P53 prevents the propagation of the genetic unstable cells and making it one of the most important tumor suppressors frequently mutated in the cancer cells. Apoptosis is also called the programmed cell death, and this slide introduces apoptosis as controlled and non-inflammatory processes, which is essential for the development and the tissue homeostasis. It maintains the balance between the cell proliferation and the cell death, ensuring that there is a proper tissue function. The apoptotic processes begins with the cell shrinkage and the loss of the surface structures, which is followed by the membrane bleaving, and internally the chromatin condenses and the nucleus fragments, and chemically collapses are activated, caspases are activated, leading to the degradation of the DNA and cellular proteins. Eventually, the cell breaks into the membrane-bound apoptotic bodies, and these are then recognized and engulfed by the phagocytes in the process called efferocytosis, which is mediated by the signals such as the phosphatidylserine exposure on the cell surface. Importantly, this clearance occurs without the triggering the inflammation and distinguishing the apoptosis from the necrosis and ensures the tissue integrity. Mechanisms and the features of the apoptosis. So morphologically, apoptotic cells develop protrusions such as the microtubule spikes and apoptopodia, which helps in the fragmentation and the packaging of the cellular contents like the apoptotic bodies. The apoptotic process is divided into the three phases. The initiation phase begins with the apoptotic stimuli, and the decision or the effector phase involves the mitochondrial disruption and releases of the pro-apoptotic factors. The execution phase is driven by the caspases, leading to the systemic breakdown of the cellular components. There are multiple signaling pathways that initiate the apoptosis. The intrinsic pathway is triggered by the death receptors, such as the FAS and TNF receptors. The intrinsic pathway is mediated by the mitochondrial signals, which in response to the internal stress, and there is also an stress response, or the endoplasmic reticulum stress pathway, that contributes to the caspase activation. All these pathways converge on common execution phase, ensuring the efficient and controlled cell death. The caspases are the cysteine aspartic proteases, and that's why they are called caspases. And they explain the central executioners of the programmed cell death, functioning as the cysteine proteases that cleave the substrates at aspartate residues. They are the synthesized and they are synthesized as inactive zymogens and are classified into the initiator caspases such as the caspase 8 and 9. There are also the effector caspases such as the 3, 6, and 7. The initiator caspases are activated through the multi-protein complexes and then activate the effector caspases via the proteolytic cleavages and create a cascade that amplifies the apoptotic signals. The effector caspases degrade the key cellular proteins, leading to the structural and dismantling of the cell and the apoptosis. Clinically, the dysregulation of the caspases contribute to the disease, and the tumor cells may evade the immune destruction by overexpressing the FAS ligand and introducing the apoptosis in attacking the lymphocytes. Conversely, the defective apoptosis signaling promotes the tumor growth and drug resistance. The mutation in the apoptotic pathway, such as the FAS mutations, can also lead to the disorders like autoimmune lymphoproliferative syndromes, also called as the ALPS. The apoptosis-inducing ligand, or the TRAIL, is a member of the TNF family that induces the apoptosis through the death receptors such as the DR4. TRAIL, or the trail, binds to the receptors, triggering the formation of the death-inducing signaling complexes that activate the initiator caspases, particularly the caspase 8. This leads to the activation of the downstream effector caspases and ultimately the programmed cell death. Interestingly, trail signaling can also activate the NFKB pathways and promotes the gene transcription survival signals. Uh, this dual role highlights the complexity of the death receptor of the signaling, and the DR4 is widely expressed in tissues such as the spleen and leukocytes, emphasizing its physiological importance. The balance between the apoptotic signaling and survival pathway determines the final cellular outcome. The mitochondria and the BCL2 family in apoptosis. This pathway of the apoptosis is regulated by the BCL2 family protein. The pro-apoptotic proteins such as the BAX and BAK and BID, promote the mitochondrial outer membrane permeabilization, while the anti-apoptotic proteins like BCL2, BCLXL inhibits this process. The balance between the opposing forces determines whether the apoptosis is initiated or not. When the pro-apoptotic signals dominate, the pores from the mitochondrial membrane, leading to a release of the cytochrome C and other factors like SMAC and Diablo. The cytochrome C binds APAF1 and procaspase 9 to form the apoptosomes, activating the caspase 9, initiating the caspase cascades. Additionally, the death receptor pathways can amplify this process via bead cleavage. This pathway is crucial in eliminating the damaged or the stressed cells. The inhibition of the IAPs, or the inhibition of the apoptotic proteins. It explains how IAPs regulate the apoptosis by directly inhibiting the caspases, thereby preventing the cell death. The proteins such as the XIAP binds to the caspase 3, 7, and 9, blocking the activity and promoting the cell survival. IAPs also participate in signaling pathways like the NFKB and the MAP kinase, contributing to the inflammation and the tumor progression. During the apoptosis, mitochondrial proteins such as the SMAC and Diablo and OMI-HTRA2 are released into the cytosol. These proteins bind to the IAPs and neutralize the inhibitory functions, allowing the caspases to become active. This step acts as a critical checkpoint, ensuring that the apoptosis proceeds efficiently, thus overcomes the IAP-mediated inhibitions and is essential for full execution of the apoptotic programs. The integrated regulation of the apoptosis, this is basically the cell fate decision, integrates the multiple signaling pathways that determine whether a cell survives or undergoes apoptosis. On one side, the pro-survival signals such as the NFKB promotes the transcription of anti-apoptotic genes like BCL2 and IAPs, inhibiting the cell death. On the other side, the cell's stress signals activate the pathways like JNKP53 and ROS, which promotes the expression of the pro-apoptotic proteins like BAX. Mitochondria serve as the central execution point where the cells converge, leading to the cytochrome C release and caspase activation. The crosstalk between the pathways such as the ROS enhancing JNK signaling creates the feedback loop that reinforces the apoptosis. If the pro-apoptotic signals dominate the cell undergoes programmed cell death, and if the survival pathways prevail, the cells may continue proliferating not abnormally. This balance is critical in cancer, where the disruption of the apoptotic pathways allows the cells to survive and continue the growth. Angiogenesis Angiogenesis is the form of the physical healing of the tumor progression, and it explains a normal physiological process where new blood vessels forms from the pre-existing ones. This is essential for the wound healing, reproduction, and the tissue repair. Under the normal condition, this process is tightly regulated by a balance between the pro-angiogenic and anti-angiogenic factors, ensuring there is a controlled vessel growth. However, in cancer, this balance is disrupted, leading to what is called the angiogenic switch, where tumors begin secreting the growth factors that stimulate the uncontrolled vessel formation. As the process progresses, the tumor cells releases the signals that activates the endothelial cells, causing the degradation of the basement membrane and enabling the endothelial cell migration and proliferation. These cells then form a new vascular structures that mature into the functional blood vessels, supplying the nutrients and the oxygen to the tumor. This newly formed vascular structure not only supports the tumor growth but also provides a pathway for the cancer cells to enter the circulation and metastasize. Tumor angiogenesis and the metastasis is a multi-step process, and this slide explains how the tumor progression relies on a stepwise angiogenesis process followed by the metastasis. Initially, the tumor cells secrete angiogenic factors that bind to the receptors on the nearby endothelial cells, activating the intracellular signaling pathways. This leads to the production of the enzymes such as the matrix metalloproteinases or MMPs, which degrades the basement membrane and extracellular matrix, allowing the endothelial cells to migrate and form the sprouts. These sprouts develop into the new blood vessels through the tube formation and stabilizes, ultimately creating a vascularized tumor. Once vascularization is established, the tumor cells gain access to the bloodstream, initiating the metastasis. The metastatic process involves the detachment of the tumor cells, entry into the circulation, survival into the bloodstream, and extravasation into the distant tissues, and formation of the secondary tumors as well. Each step is tightly regulated but often becomes highly efficient in aggressive cancers. Now, the routes of metastasis can be of the four different types, four primary pathways, and this slide outlines the four main routes which can cause the spread of the cancer to the distant site. The first is the transcoelomic spread, where the cancer cells disseminate across the body cavities, such as the ovarian cancer, seeding with the peritoneal cavity. The second and the most common is the lymphatic spread, where the tumor cells travel through the lymphatic vessels to the regional lymph nodes and eventually enters the systemic circulation. The third route is the hematogenous spread, involving the invasion into the blood vessel, particularly the veins, allowing the tumor cells to travel to the distant organs like the lungs or the liver. And the fourth route is the transplantation or the implantation, often occurs during the medical procedures or the local spreads to the nearby tissues. These pathways collectively explain how the cancer progresses from the localized tumor to the widespread systemic diseases. Proangiogenetic factors are the key regulators of the tumor angiogenesis, and this slide focuses on the molecular drivers of the angiogenesis, particularly the VEGF family, angiopoietin, and ephrine signaling systems. So the VEGF acts as the primary angiogenic cytokine, binding to its receptors on endothelial cells to promote the proliferation, migration, and increased vascular permeability. This makes the VEGF a central driver of the new vessels, which forms the tumors. Angiopoietins, on the other hand, regulate the vessel stability, where ANG1 promotes the maturation and the stabilization, where ANG2 destabilizes the vessels, making them responsive to the VEGF-driven angiogenesis. The ephrin and the EPH receptors interaction further regulate the communication between the endothelial and the surrounding cells. This contributing to vascular patterning and remodeling. Together, these factors coordinate to complex processes of the vessel formation, maturation, and adaptation of the tumors. The TSP1 and the Notch signaling in angiogenesis and cancer. So this slide highlights two important regulatory systems in angiogenesis, the TSP1 and the Notch signaling. The TSP1 is a glycoprotein which modulates the endothelial cell functions such as the adhesion, migration, and the proliferation, often acting as an anti-angiogenic factor. It helps maintaining the balance by counteracting the excessive vessels and formation of the vessels. The Notch signaling, on the other hand, is a highly conserved pathway that regulates the vascular development and the endothelial cell behavior. The activation occurs through interactions between the Notch receptors and the ligands like uh, delta and jagged, influencing the cell fate decisions. In the cancer, the dysregulation of the Notch signaling leads to the abnormal vessel formation and the tumor angiogenesis. Because of its central role, targeting the Notch signaling is being explored as a therapeutic strategy to inhibit the tumor vascularization and progression. The vascular endothelial cell interaction and the survival signaling explains how the endothelial cells maintain the structural integrity and survival through the both cell-cell and the cell matrix interactions. The VE cadherin, or the vascular endothelial cell interactions, mediated cadherin mediated adherence junctions form the tight connections between the endothelial cells, creating a mechanical barrier that regulates the vascular permeability. This simultaneously integrates the interactions with the extracellular matrices or ECM, activating the focal adhesion complexes involved in the proteins like FAK, SRC, Paxillin, and CAS, which transmits the survival signals into the cells. In addition, the growth factors such as the VEGF, angiopoietin 1, activate the intracellular vascular development and endothelial cell behavior. The activation occurs through the interaction between the Notch receptors and the ligands, like the delta and the uh, jagged, influencing the cell uh, fate. In cancer, the dysregulation of the Notch signaling leads to the abnormal vessel formation and tumor angiogenesis. Because of its central role, the targeting Notch signaling is being explored as therapeutic strategy to inhibit the vascularization and the progression. The interleukin and the interferons. They act as the anti-angiogenic and the anti-tumor mechanisms, and it highlights like the cytokines, how the cytokines like interferons IFN alpha and IFN beta, as well as interleukins IL-12, they inhibit the tumor angiogenesis and the growth. IFN alpha suppresses the VEGF transcription by inhibiting the transcription factors such as the SP1 and the SP3, thereby reducing the angiogenic signaling at the genetic level. Additionally, IFN alpha combined with the retinoic acid shows the synergistic effects, significantly inhibiting the vascular tumor vascularization uh, and the growth in the experimental models. IFN beta primarily exerts its anti-tumor effects by reducing the tumor blood supply through the angiogenesis inhibition rather than directly being cytotoxic. The IL-12 stimulates the immune cells like the T cell and the NK cells or the natural killer cells to produce the IFN gamma, which further suppresses the angiogenesis. And this mechanism demonstrates that immune signaling pathway can effectively target the tumor vascularization and the growth. The role of epigenetic modifications in cancer. So basically, epigenetic changes regulate the gene expression without altering the DNA sequences, primarily through the histone modification and the DNA methylation. In normal cells, the euchromatin allows the gene expression due to the relaxed chromatin structure, whereas the heterochromatin is tightly packed and transcriptionally active. In cancer, the abnormal histone modification disrupts this balance, leading to the inappropriate gene activation or silencing. The DNA methylation plays a critical role where the hypermethylation of the promoter regions silences the tumor suppressor genes like the P16, BRCA1, and MLH1. Conversely, the hypomethylation leads to the genomic instability and activation of the oncogenes such as the CMYC and HRAS. Together, these alterations contribute to the tumor initiation and progression by the disruption of the normal gene regulation and cellular homeostasis. Histone modifications are the regulations of the gene expression and the chromatin structure. Different types of the histone modifications and their functional consequences on the chromatin structure and the gene expression are demonstrated here. The histone methylation can either activate or repress the transcription depending on the specific residues modified, such as the H3K4ME3 activation or H3K9ME3 via repression. The histone acetylation mediated by the HATs relaxes the chromatin and promotes the gene expression, whereas the deacetylation by HDAC condenses the chromatin and represses the transcription. The histone phosphorylation is involved in the process like the transcription, activation, DNA repair, and the cell division. This highlights its dynamic regulatory role. Additionally, there is a significant crosstalk between the histone aberrations and the DNA methylation, forming a complex regulatory network where the disruption of these processes in the cancer leads to the aberrant gene expression, contributing to the tumor development and progression. Now let's coming to the oncovirus induced cancer. So the mechanisms of the oncovirus induced cancer is being demonstrated here, how oncogenic viruses contribute to the cancer through multi-step processes. Initially, viruses establish the persistent infection either by integrating into the host genome or remaining episomal. They then express the viral oncogenes that interfere with the host regulatory pathways, promoting the uncontrolled cell survival and the proliferation. A key step involves the activation of the tumor suppressor, such as the P53 and the RB protein, leading to the loss of the cell cycle control. So viral proteins such as they also activate the proliferative signals like the MAPK and PI3KKT, as well as the JAK-STAT, while inhibiting the apoptotic process through the proteins like the BCL2. And over the time, the accumulation of the mutations and the genomic stability leads to the clonal expansion of and malignant tumor formation. So distinguishing the characteristics of the oncoviruses. This slide summarizes the unique features that diminishes the oncoviruses in the cancer development. One key characteristic is a long latency period where the infection may persist for years before the cancer development, whereas acquiring the whereas it requires the additional genetic or additional environmental factors. Although the viral infections are common, the cancer development is relatively rare and indicates that the infection alone is insufficient for transformation to cancer cells. The oncoviruses encode the specific oncogenes that targets the host regulatory pathways rather than causing the random damage. They often show cell type specificity, infecting the particular tissues such as the epithelial cells or the lymphocytes. Additionally, the viral persistence and the host immune status play critical roles, and as immunocompromised individuals are at the higher risk. These characteristics highlight the complex interplay between the virus and the host cells in the cancer development. So the DNA oncoviruses and the RNA oncoviruses are the two classes of the oncoviruses. DNA oncoviruses can be classified further into the small and the large DNA oncoviruses. Some examples of the DNA oncoviruses are the adenoviruses, SV40 human papilloma viruses, whereas the herpes viruses are the large DNA oncoviruses like EBV, Epstein-Barr virus, or the Kaposi sarcoma herpes virus. So other hepatitis viruses B are also present. In RNA oncoviruses, the hepatitis virus C is one of the RNA oncoviruses, whereas the leukemia T cell leukemia virus is another RNA oncovirus. The characteristics of the RNA oncoviruses are that they are associated with the neoplasms, that is the adult T cells, leukemia, and the lymphoma, and some cases with the hairy cell leukemia. And another one is associated with the neoplasm or the lymphoma. The cancer stem cells are the small subpopulation of the tumor cells with stem-like properties such as the self-renewal and the differentiation. So the CSCs drive tumor growth by uh, generating both identical stem cells and diverse non-tumorigenic cells, contributing to the heterogeneity and clinical heterogeneity. So the CSCs clinically are highly significant because they contrast two models, the CSC model or the hierarchical organization and the clonal evolution model, which is the mutation-driven diversity. The evidence from the cancers like leukemia, glioblastoma, and the breast cancer supports the existence of the cancer stem cells, making them the key therapeutic targets. Talking about the clonal evolution, which is basically the tumor heterogeneity and the progression, the tumors evolve from a single mutated cell through the accumulation of the genetic alterations over the time. As the mutations accumulate, the different subclonal emerges, each with varying survival and the growth advantages, leading to the tumor heterogeneity. Some subclones become dominant due to the selective advantages such as the faster growth or the therapy resistance. The two patterns are described here, which is the linear evolution and branched evolution. The tumor microenvironment further influences the selection pressures, promoting the aggressiveness and treatment-resistant cancer populations. So, hypothesis of the origin and the isolation strategies of the cancer stem cells. The major hypothesis explaining the origin of the CSCs is the stem cell mutation, adult stem cell transformation, and the de-differentiation of the mature cells. In the stem cell mutation hypothesis, in the stem cell mutation hypothesis, the mutations in the normal stem cells are inherited by progeny, increasing the cancer risk. The adult stem cells hypothesis suggests the long-lived stem cells accumulate mutations over time due to the frequent divisions. The de-differentiation hypothesis proposes that the mature cells revert to a stem-like state after acquiring the mutations. This slide also outlines the methods for the CSC identification, including the FACS using the surface markers, the site population assays based on the dye efflux, and the sphere forming assays. The functional validation is performed using a in vivo transplantation into the immunodeficient mice, demonstrating the tumor initiation and the self-renewal. These techniques are crucial for studying the CSC biology and developing the targeted therapies. Therapeutic strategies targeting the cancer stem cells through the key signaling pathways and the telomere biology, the cancer stem cells can be targeted. So pathways such as the W-anti-beta-catenin, hedgehog, and the notch, they regulate the cancer stem cell maintenance and the survival. And their inhibition can induce the cancer stem cell populations. For example, the cyclopamine inhibits the hedgehog pathway while gamma secretase inhibitors block the notch signaling, reducing the tumor growth. Additionally, the cancer stem cells rely on the telomerase activity to maintain the short telomerase, making the telomerase a therapeutic target. The telomerase-based immunotherapy, such as the GV1001 peptide vaccination, stimulates the immune responses against the cancer stem cells. These strategies aim to eliminate the cancer stem cells, specifically improving the treatment outcomes and reducing the relapse. The overall combining pathway inhibition with the telomere targeting offers a promising approach for the durable cancer therapy. This slide highlights how the cancer stem cells depend on the specialized microenvironments known as the niches for survival, self-renewal, and the differentiation. These niches include the signals from the tumor vasculature and the VEGF, and hypoxic conditions mediated by the hypoxia-inducible factors or HIFs. They support the maintenance and the tumor progression. The therapeutic strategies such as the anti-VEGF antibodies, uh, bevacizumab, and HIF inhibitors aim to disrupt these external signals, thereby weakening the CSCs or the cancer stem cells and their survival and reducing the tumor growth. By By targeting both intrinsic and the extrinsic regulatory pathways, these approaches can be effectively deplete the CSC population and limit the recurrence. The second part explains the induced potent stem cells or the iPSCs, where the differentiated somatic cells are reprogrammed into the pluripotent state using the Yamanaka factors, and these iPSCs behave similarly to the embryonic stem cells. The processing of the self-renewal and the differentiation capabilities. This breakthrough first demonstrated in 2006 and later awarded the Nobel Prize for the revolutionary regenerative medicine in the cancer research. The iPSCs are also provide the valuable models to study the cancer initiation and the cancer stem cell behavior. So the role of the animal models in cancer drug discovery. Starting from the drug design to the in vitro screening to critical evolution stages, it requires the in vivo validation. While the highly throughput screening in the cell lines is efficient, it lacks the full physiological relevance, making the animal models essential for confirming the therapeutic efficiency. So the steps in the drug discovery, evaluating the pipelines and evaluation pipelines are the after drug discovery, it goes through the high throughput screening, followed by the evaluation, which is the critical stage, and improper method selection may drop the potential antigens in vitro, and in vitro has the limitations. So the criteria for in vivo model selection is that ideal model characteristics should be close representation of the human disease, whereas the feasibility of the large scale screening and it should be economical. Uh, the balancing factor should be there should be clinical relevance and the practical cost. And the classification of the animal cancer models can be spontaneous tumor models, whereas the induced tumor models. Both are the transplantable tumor models with different types like heterotrophic, orthotopic, and then syngenic, and then xenogenic. These are the induced in the genetically engineered mice or GEMs, and there are the two different types, which are the transgenic animals as well as the knockout animals, where in transgenic animals gene insertion takes place and gene deletion takes place in the knockout animals. Now, why use of these models? They prevent the diagnosis and treat the human diseases, and they are physiologically complex, and they are cost effective. And why these mice is specifically used is they are small and easy to handle. They have the short generation and the short lifespan. They are anatomically and physiologically similar to the humans with 95% genome similarity to humans. They have the shared genes for the complex diseases and the genome directly manipulatable. So what are the spontaneous tumor models in cancer research? So basically these models allow the study of biological history of the natural diseases, and they are available in cancer model, and they can be spontaneous. They are virus induced transgenic knockouts and induced carcinogens, and as well as transplant and planted. The spontaneous models have general characteristics, whereas the spontaneous tumor models have the specific examples and the features. uh, there are some limitations and the challenges, like impractical to obtain the sufficient comparable numbers for screening. They are measurable only in the late metastasis and are non-uniform. Difficult to establish accurate and staging, and not reproducible, and they may have the viral origin. The additional pro and the plus points is that they provide a stringent test of the anti-tumor activity. There are certain virus-induced tumor models which are used in the cancer research, And this slide explains the tumor models generated using the oncogenic viruses such as the uh, Fried leukemia virus and the mouse memory tumor virus and the Rous sarcoma virus. These models help us in understanding the viral oncogenesis, including the mechanisms like the insertion, mutagenesis, and the activation of the cellular oncogenes. For example, the MMTV can integrate into the host DNA and activate the oncogenes like the ABL or RAF, leading to the tumor formation. These systems have been critically discovering the fundamental cancer-related genes. The induced tumor models are also used in the cancer research, which are basically the radiation and the chemical carcinogens. And this slide explains the experimentally induced tumor models using the radiation or the chemical carcinogens to initiate the cancer. The radiation-induced tumor models, particularly the skin cancer, are useful due to the easy accessibility and the observation, but they involve the safety and hazards and long induction times. So chemical carcinogen models such as the DMBA, TPA are the two-stage skin tumor models closely resemble to the human cancer development and allows the controlled study of the tumor initiation and promotion. These models are widely used for the studying of the carcinogenic mechanisms and to test the chemopreservatives. Chemopreventive agents. So certain chemical carcinogens and transplantable tumor models are also used in the cancer research. These are the specific chemical induced carcinogenesis models like the DMB induced rat and then the DMB induced colon tumors and which has certain limitations. Then there are the MNU induced tracheal squamous cells, and then there is the DEN induced lung adenocarcinoma. So these are used in the various species with different dosages of the carcinogens, and the results are analyzed over a period of time. The summary of the table of additional chemical models are listed here with the colon, prostate, esophagus, and the breast cancer types. So there the different species are used and the different carcinogens are used. There are the transplantable tumor models. So based on the transplantable cancer cell lines or the tissues into the host animals, they can be mice or the rats as per the choice of interest. And they can be heterotropic transplantation or the orthotropic transplantation. So a heterotropic transplantation is where the tumor cells are implanted at the site different from the tissue of the origin. Example, the subcutaneous. Whereas the orthotropic transplantation is where the tumor cells implanted into the corresponding tissue of origin for better mimicking the natural microenvironment. So the transplantation models in the cancer research has different methods, sources, and applications. The core concepts and the advantages is that the transplantation models, like the tumor cells or the tissues, like they are transplanted into the host mouse, and they can be different types like ectopic, heterotopic, or orthotropic. And the advantages are they are typically cheaper and the faster to use and not covered by patents. The application is commonly applied in the studies of the genetics and the diagnostics. Then there are the tissue sources and implant sites. Based on this, there can be syngenic, allogenic, and xenogenic. And the implant sites can be orthotropic or the heterotropic. Then the heterotropic transplantation, basically they are the common sites and the forms for the intracranially or subcutaneous types. And then we have the orthotropic transplantation and the methods, which defines that the transplantation of the cancer cells to the anatomic location and the tissue of the origin. The advantages are they have the better mimicry and more close resemblance to the human cancers in terms of the histology, vascularity, gene expression, as well as the chemotherapy response and the metastatic biology. The methods are the direct injection as well as the surgical orthotropic implantation. Now, talking about the human tumor xenografts in the cancer research, there are the four different. This has been divided into the four different stages. So the general transplantation models has the certain limitations. So histopathology cannot be exactly correlated to the human tumors, and there is a lack of the stepwise progression through the pre-neoplastic stages. The evolution of the tumor cells during the passaging is a challenge. Then it requires for the angiogenesis to support the newly transplanted tumor. So the on contrary, the euthymic nude mouse model was developed in 1960 with mutation in the NU gene on the chromosome 11. The phenotype is retarded growth and low fertility with no fur and immunocompromised. So the key feature is that it has the lack of the thymus gland and the T cell immunity is a challenge. So the first xenograft was in 1969. The first human tumor xenograft with the colon adenocarcinoma was conducted, and the methodology is that it has the endpoint and sites endpoint and analysis. So basically xenograft sites can be subcutaneous, intraperitoneal, then intracranial, which is basically inside the brain, and then it has the intrasplenic, then renal subcapsule, and the site specific or orthotopic. And the study endpoints are the toxicity, the drug-related deaths, and the net animal weight loss can be monitored. And the efficacy is that the tumor weight challenge changes, and the treated versus controlled survival ratio and the tumor growth assay are corrected for the doubling time. Advantages and the disadvantages are listed here, like many human tumor cells, like cell line transplantable, and wide representation of the most human solid tumors, as well as the good correlation with the drug regimens active in the lung, colon, and the breast cancer or the melanoma are some of the advantages. Disadvantages is that the brain tumors are difficult to model, and the differential behavior metastases are rare. Survival is not ideal endpoint, whereas the shorter doubling time than in humans, and they are difficult to maintain due to the risk of the infections. The transgenic and the knockout mouse models and are in vivo hollow fiber assay in the cancer research. So the oncogene expressing transgenic models, and there are the knockout mice models, and there is in vivo hollow fiber assays. So these are the three different types of the like models in the cancer research. The oncogene expressing transgenic models are basically the different types like RAS mutation model, then the TRAMP transgenic mice, and then the P53 WNT1 and the APC models. Different types of the knockout mice models can be NKX 3.1 knockout, and the homozygous P53 knockout, and the BRCA1 conditional knockout. Different types of the in vivo hollow fiber assay are the cell lines, and then the implantation, and then treatment and the analysis and the endpoint. And these are the different steps in the in vivo hollow fiber assay. The evolution and the utility of the cancer models in the drug discovery and the research can be concluded as they are the historic primitive models. They are induced tumor models with the chemical radiations. Then there are the modern screening and the disease-oriented approaches, and they are the genetically altered models and conclusions. So let's discuss the cancer screening techniques. What is cancer screening? So cancer screening involves in a lot of the techniques such as the PET, CT scan, MRI scans, and the blood tests. So although the screening techniques are quite reliable, there are the problems of the false negative tests, so where a false positive test where cancer is detected in a normal person who actually does not have the disease with cancer and is declared as normal, leading to the delay in the diagnosis and effective treatment. An overdiagnosis is a condition where the disease or where the diagnosis of a condition may not have been clinically significant and had not been detected in screening. So cancer screening is a powerful tool for early detection, but it's not perfect. The false negatives delay treatment, false positives causes the unnecessary anxiety and interventions, and the overdiagnosis leads to the overtreatment of the clinically insignificant disease. Therefore, modern screening strategies increasingly focus on the risk-based approaches, improved molecular diagnostics, and balancing the benefits against the potential harms. So the first one is the blood test or the alpha fetoprotein test, where the AFP is a protein which is made by the developing fetus, and the concentrations are highly in the blood when the baby is born and fall to low level at the age of the one. High level of this protein in the blood can be the marker of the liver cancer and the cancer of the ovaries or the testicles. However, high AFP concentrations are also indicative non-cancerous liver, such as the liver disease, such as the cirrhosis and hepatitis, and must therefore be done in conjunction with other diagnostics and the screening methods. So AFP is used as a monitoring tool with decreasing levels and indicate the response to the treatment. If the concentration after the cancer treatment do not significantly decrease, usually to normal or near normal levels, then some of the tumor tissues may still be present. These concentrations are measured using the ELISA tests, and the AFP test is not highly specific or selective, and not every individual with cancer may show the elevated levels of alpha fetoprotein. Then there are the prostate specific antigens or the PSA test, which detects, monitors, and improves with the uh, and has an improved accuracy. The measures it measures the protein produced by the normal and the malignant prostate cells, and elevated levels indicate the potential cancer and the other conditions. So this screening is measured in the nanogram per ml via the ELISA or the enzyme-linked immunosorbent assays, and used for monitoring the disease progression and the treatment response. Certain limitations are that the elevated PSA can also be caused by the prostatitis or the inflammation, as well as the benign prostate hyperplasia or the BHP, which is basically the enlargement of the prostate glands. The test is used in combination with the digital rectal examination for better accuracy. Now, the improving test accuracy advanced in the matrix is that the free versus the total PSS, where the free PSA and the bound PSS are calculated, and the proportion of the free PSA is calculated. If there is lower, then the proportion of the free PSA associated with more aggressive cancer. So the PSA test is a valuable but not a definitive tool, and combined with the DREs and refined matrices, that is the free versus the total ratio, and the density age specific ranges, and the diagnostic accuracy and the risk stratification are significantly improved for the prostate cancer management. Then we have the CA125 tests, which is basically used in the PCOD. The measurement amount of the protein, the CA125 or the cancer antigen 125 in the bloods, the higher levels indicate that the benign condition of the ovarian, endometrial, peritoneal, or fallopian tube cancer, whereas the other test performed to determine the accurate diagnosis, and the CA125 levels measured in the units of the units per milliliter, and the normal value is 46 units per milliliter. So the clinical uses and the applications is the monitoring the treatment and the screening of the high risk individuals and detecting the recurrences. And it also has certain limitations and disadvantages, which is not for general screening. It has low specificity, low sensitivity, and uncertain benefits. Now there is another test, which is the fetal occult blood test or the FOBT test, and it is used for the detection of the occult or the hidden, that is the blood in the stool samples. The primarily for early detection of the colon cancer and precursors. The purpose is to early detection of the colon cancer, as well as the detection of the precursor polyps that breed slowly, and it's done before the symptoms appears. So the advantages is that it is an inexpensive and easy to do. It has an early detection, and it helps to avoid the metastasis. So it uses the antibody of the gold complex solution and targets the globin, and the antibody binds to the globin complex and settles as the visible line. So a positive result means the test line uh, in the test line shows a positive result. The advantages of the immunogenic FIT over the chemical, like GUIC, is that uh, so basically guaic is one of the chemical testing, which is basically uses the guaic or oxidizing agent and the solution and it attaches with the hemoglobin, heme particle of the hemoglobin, and oxidizes with guaic. So heme and the oxidizer basically the makes it the guaic, and it turns it visibly blue. So blue color means a positive test. So it has the higher sensitivity and the higher specificity. Then we have the physical form of the detection, which is the ultrasonography in cancer diagnosis. So it is a fundamental and it has the mechanism. Fundamental mechanism is that the ultrasonic pulses of the more than 20 kilohertz are being subjected on the tissue or the site of the cancer, and when it echoes back, it gives an image or the impression of the tissue or the cancer or the solid mass which is formed. The key features are the advantages like real-time imaging. It's portable and low cost, and doesn't have the challenge of the ionizing radiation. However, the disadvantage is that it's operator dependent and limited field of the view, and it requires skilled interpretation. The clinical applications in oncology for ultrasonography is for the thyroid, head and the neck, then the pulmonology and general oncology. The advanced techniques are the contrast enhanced ultrasound or the CUES, CEUS, and parametric imaging. So here the vascular signatures are characterized with parametric image, which is basically color coded. Now then we have the CT scan or spiral computed topography, or the spiral CT, along with the low dose CT or the LDCT. So the spiral helical or the CT scan has the principles and the advantages. Like the mechanism is continuously rotation of the X-ray source and detects while the patient table moves, acquiring the data in the spiral path. Whereas the LDCT is a technique which combines the specialized X-ray equipment with sophisticated computers using the significantly less radiation. Then we have the PET CT scan, which is an integrated metabolic and anatomical imaging for cancer. So basically, the CT scan along with the PET scan is forms the along with the PET scan forms the PET CT scan, which combines both detailed function and the structural information. So basically, radioactive tracer injections levels the sugar are swallowed, inhaled, or the injected in the patient, and this accumulates in the areas where there is a high metabolic activity. And then the PET CT scan detects the tracer emissions like the metabolic activity. Then the computer tomography takes the X-ray images and anatomical structures, and the PET image is the bright, whereas the CT image is the detailed. So and this combinedly focuses the metabolic hotspots and localized on the anatomy. Then we have the colonoscopy, which is the preventative screening and the diagnostic tool. It detects and removes the abnormal colorectal growths of the polyps to prevent the cancer. And there are the different types of the colonoscopy, like the standard, which is the optical colonoscopy, whereas the virtual colonoscopy is with the assistance of the CT colonoscopy. The optical colonoscopy has the different procedures where the insertion and the inflammation of the colonoscopy is inserted, like with air or the carbon dioxide, to expand a clear view. Then the examination of the entire colon and the rectum is done, like the abnormal growths can be removed, and samples can be uh, collected via biopsy. The advantages is that it's highly sensitive and one of the most effective screening tests, and views the rectum and entire colon, and allows for the immediate biopsy or spectromy during the procedure. The disadvantage may sometimes miss small and the flat depressed lesions, and requires thorough bowel preparation like cleaning and diet changes. The small risk of the bleeding and the tearing and the perforation are also one of the disadvantages. There is the digital rectal examination, which is a process to detection of the prostate cancers in the men and ovarian or the uterine cancers in women. So to perform this, the doctor will gently insert the gloved, lubricated finger into the anus, and this allows them to feel for any abnormalities. For example, An enlarged prostate may feel like bulged behind the rectum wall, and prostate cancer may feel like bumps or normally smooth. So there is again flexible sigmoidoscopy, which is basically the evaluation of the lower large intestine colon cancer screening, and the procedure is the insertion of the thin flexible tube, which is basically the sigmoidoscope into the uh, rectum, which has a tiny video camera, and the tip allows the viewing of the rectum and most of the sigmoid colon. There is mammography, which is basically used for the breast screening. So basically early detection tools using the x-ray to visualize the breast tissues, identifying the approximately 25% of the cases, and the overviews and the signs of the breast cancer. The some of the signs like lumps, breast pain, nipple discharge, and the thickening of the skin, changes in the cell in the cell size and the shape are the early signs of the cancer. The screenings is a routine, like the no woman with no symptoms, routine detection of the breast cancer, and like for the symptoms, they use the suspicious screening for the symptoms with more details. The reliability of uh, this screening technique is that the tumor is easier to spot, uh, whereas for the dense tissue, the tumor is hard to find and appears white on white. The mammography is less reliable for women with the dense breast tissues and more common in younger women. And it's both the tumors and dense tissue appear white. Then there is the MRI, which is basically the magnetic resonance imaging. And the principle of this is like it uses the powerful magnets and the radio waves to produce the computer generated images and measures the tumor size and can distinguish benign and the malignant tumors. So clinical applications are like rectal and the prostate cancer, neurological cancers, and the other tumors in the body. And MRI can also be used for the breast screening. And there are certain safety considerations like pregnancy and ionizing radiation and general safety should be taken care. The most uh, like precision oncology testing is the DNA testing. So the information they provide is instrumental and in selecting the best targeted therapies for patients' unique form of the cancer. The genetic target testing is sometimes also referred to as the genomic testing and the genetic screening and the diagnostic testing. So it detects the changes of the mutations in the DNA of the cancer cells, providing the crucial information about what mutations are present in the patient's genes. It helps the treatment team to select the best possible targeted therapy and can predict which treatment will be benefiting the patient the most. There are the liquid biopsies as well and the HPV testing. The liquid biopsy, a sample of the patient's blood is obtained through the sample blood draw and which simply is then tested for a short list common mutations. And that turnaround time is very fast and typically less than a week. HPV testing is for the detection of the cervical cancer prevention, and it detects the HPV DNA and like human papilloma virus DNA. And the cell collection is done via the speculum and brush, and it identifies the risk of the abnormal cells and the cancers. It can be performed with the PAP test and is uh, for the both of both the different genders. Then comes the microarray tests, so which is a two-dimensional array on the solid substrate and is usually uses a glass slide of the silicon thin film cell that assess the tests. So large amounts of the biological materials using this high throughput screening miniaturized, multiplexed and parallel processing detects the uh, uh, and uses these detection methods. So there are the different types of the microarrays like the DNA microarrays such as the cDNA microarrays, oligonucleotide microarrays, and the BAC microarrays and SNP microarrays. Coming to the microscopy and the immunohistochemistry in cancer diagnosis. So microscopy uses the pap smear to analyze the cervical cancer screening, whereas in immunohistochemistry, peroxidase and the substrates are used in the enzyme linked immunosorbate assays, and using the fluorophore tagged secondary antibodies. This shows the exact protein localization within the tissues. So there are other diagnostic and the research applications based on the biomarkers, and these biomarkers can be cytokeratin, then CD117, CD10, and estrogen and progesterone receptors, which are applicable in different types of the cancers and the tumors. Then we have the chemotherapy, which uses the chemical agents to destroy destroy and inhibit the growth of the cancer cells. It originates the date back to the World War II, when the observations from the autopsies of the soldiers exposed to nitrogen mustard revealed the profound bone marrow suppression and the lymphoid tissue depletion and the gastrointestinal uh, ulceration. So it derives the developed derivatives developed as the anti-cancer agents, and this is used in the modern oncology. So the mode of action is that it targets the M phase and the S phase. So these cell cycle specific primary targets are there, which the cells with the high division rates, they target the cells with high division rates, and there are anti-metabolites and the vinca alkaloids, whereas alkylating agents are the cell cycle non-specific uh, targets. So there are the different drug classes like the alkylating agents, antimetabolites, vinca alkaloids, antaxanes, and the antibiotics, which target the different phases of the cell cycle. And the drug effectiveness factor is the tumor growth factor, doubling time, and the concentration of the drug and the exposure duration. Now, the classifying the chemotherapeutic agents are of the two different types. One is the cytotoxic drugs, and another is the hormonal agents. Cytotoxic drugs can be alkylating agents, and then antimetabolites, platinum compounds, plant alkaloids, and anti-tumor antibiotics, and miscellaneous agents. So there are some of the examples of these different antimetabolites, whereas the hormonal agents are the effective hormone-dependent cancers such as the breast and the prostate cancer. Some of them are glucocorticoids. Uh, so prednisolone is for the glucocorticoids, and diethylstilbestrol is for the estrogens. Taxomifican, tamoxifen is for the anti-estrogens, and then the testosterone is for the androgens, and hydroxyprogesterone is for the progestins. So molecular mechanisms of the cell cycle and the uh, chemotherapeutic agents is that the cell cycle and the mitosis mechanisms and the drug targets, where they target the interphase, prophase, M phase, and the anaphase. All the different uh, cell phases are there, cell cycle phases are there, and the vinca alkaloids and the taxanes, basically they target the metaphases. And there are the other inhibitors which basically targets the RNA primer and the topo isomerases, and alkylating agents causes the DNA damage and cross linking DNA. So basically the mechanism of action of the anti-cancer drugs at the molecular level are listed here, where the methotrexate basically they inhibit the folic acid metabolism and blocks the purine and the pyrimidine system. The markaptopurine interferes with the purine metabolism, and 5-fluorouracil inhibits the thymidine synthesis. Hydroxyurea inhibits the ribonuclease nucleotide reductase, and certain alkylating agents like cross-linking DNA damage and mitotic disruptions focus on the preventing cell divisions and mitosis. So the anti-metabolites and the alkylating agents in the chemotherapy has the different mechanisms, toxicities, and specificity to the drugs. So alkylating agents, anti-metabolites, and there are the specific anti-metabolite drugs like the methotrexate, markaptopurine, and the 5-fluorouracil. They all have the different mechanisms, and some of them have the side effects and dosage limitations. Now we have the radiation therapy for the healthcare professionals. So basically, it is an effective tool for treating the cancer, and the patients are diagnosed within the cancer receiving radiations. Uh, the radiation oncologists manage the care for cure and palliation. So basically, physical and the biological basis of the clinical applications is that management of the cancer for the cure of palliation, and it has the energy deposition and the DNA damage and the cell death. So the treatment process is basically the simulation using the imaging for localization on the CT, localization using the CT scan, and then it has the treatment planning like dose calculations and the optimization. The delivery of the radiation is the targeted treatment. The types of the radiations available are the gamma radiations, X-ray radiations, photons, neurons, and the electron are the particle beams that are uh, the photons are the gamma radiations and the X-rays. The protons, neutrons, and the electrons are the particle beams. So the biological basis and the mechanism of the radiation therapy is that when a radiation beam is exposed to the DNA, the DNA damage causes the uh, breaks of the DNAs. So the normal cells have the effective repair, while the cancer cells have the impaired repair, and as a result, the dysfunctional enzymes and the enzymes accumulate the damages, and therefore the cell death and the apoptosis and the necrosis can be initiated damaging the radiation, by damaging the DNA using the radiation. On the other hand, there is the fractionation and the 4 hours of the radiobiology, where the fractionation is the dividing total dose into small fractions, exploiting the differential repair. So this has the repair, redistribution, repopulation, and the reoxygenation. So it has the certain goals and development processes. It is curative and the palliative, and the delivery is painless because external beam treatments typically 5 days a week or 1 to 10 weeks, for 1 to 10 weeks, and there are certain side effects like temporary skin redness, fatigues, and usually resolve over a few weeks. So the common side effects of the radiation therapy in the oncology side is the swelling and the skin redness in the breast cancer, and nausea and vomiting and diarrhea for abdomen. For the chest, there is the cough and the shortage of the breath and esophageal irritation. For the head and the neck, there is the taste alteration, dry mouth, and mucositis, and skin redness. For the brain, there is the hair loss and the scalp redness, and diarrhea, cramping, and urinary frequency, and the vaginal irritations in the pelvic sides. And for the prostates, it's in impotency and urinary symptoms and the diarrhea. So the radiation therapy has the following treatment processes. So basically, the first is the referral and the consultation, then the simulation using the CT scan to detect, and then the treatment is planned by the physicians in their organs at the risk, and the software using the different softwares and the algorithms with dosage calculations and medical physicist. And after that, the quality assurance and the safety is uh, taken into account, and then there is the treatment delivery, where the external beam and the internal uh, radiation therapy are also done, and this requires the follow-up and the monitoring. So discussing about the immunological approach for the cancer immunotherapy, the cancers are treated by harnessing the patient's own immune systems to recognize the and eliminate the malignant cells. For example, the cytotoxic T lymphocytes, natural killer cells, and the beta lymphocytes and the antibodies, the CD4 helper T cells, and the dendritic cells and the macrophages. The evidence supporting the immune responses against the tumors are the histopathological and the clinical evidences. There are the experimental evidences like the mouse models. So this slide basically shows the mechanisms of the immune response to the tumors. The CD8 plus or the cytotoxic T lymphocyte or the CTLs, the tumor antigen presentation occurs via the antigens and the presenting cells or the APCs. The tumor antigens are released, the APCs ingest the process and ingests and processes the antigens, and cross presentation activates the CD8 plus T cells, and the specific tumor CTLs differentiate. The CTLs recognizes the tumor cells via the MHC1 and induce the apoptosis, and this process is referred to as the induction and the effector phase of the anti-tumor response. The antibody-mediated immunity and the tumor-bearing hosts may generate the antibodies against the tumor antigens. And the mechanisms include the complex activation, antibody-dependent cellular cytotoxicity or the ADCC, then the delivery of the toxins-linked antibodies, and then the antibodies can also be engineered to carry the cytotoxic drugs or the radioactive isotopes. So there are the natural killer cells or the NK cells, which acts as the lyse tumor cells with low MSC1 expression and functions via the ADCC. They release the TNF beta and the cytotoxic factors and are stimulated by IL-2 and IL-12 interference. Macrophages are activated macrophages via the IFN gamma, and they secrete the lytic enzymes and produce the nitric oxides or NO. They release the TNF alpha and the tumor necrosis, and these mediations contribute directly to the tumor cell killing. So the forms of the cancer immunotherapy can be classified as non-specific immunotherapy, followed by the specific immunotherapy. Again, specific immunotherapy can be active immunotherapy or the passive immunotherapy. The non-specific immunotherapy are of different types like bacterial extracts, then cytokines, endotoxins, and the chemical adjuvants. The specific immunotherapy can be active immunotherapy and the passive immunotherapy. Different types of the active immunotherapy are the tumor vaccines, cytokine enhanced vaccines, DNA vaccines, viral vectors, and dendritic cell based vaccines. Whereas the passive immunotherapy can be monoclonal antibodies, adaptive T cell transfers, and the immune RNA. There will be specific therapeutic modalities like the cytokine therapy, like interleukin 2, and then the tumor necrosis factor TNF, and the IL-12 and the IL-16, as well as the GM-CSF. There are again the adaptive cellular therapy where the tissue isolates the TILs, which is followed by the expansion in the in vitro with IL-2 and reinfusion into the patient, which results into the tumor regression in some cases. So some different types of the tumor vaccines are the killed tumor cell vaccines, purified tumor antigens, and the professional antigen presenting cells based vaccines. Then there are the cytokine enhanced vaccines, DNA vaccines, and the viral vector vaccines. And the clinical applications can be melanoma, colon cancer, renal cancer, sarcoma, prostate cancer, and the beta cell lymphoma. The next is the chimeric antigen receptors or the CAR T cells. The overview and the concept is that the CAR T therapy represents a form of the engineered adaptive immunotherapy, whereas the modified T cells can be directly used for the cytotoxicity on the tumor cells. And there are the different structure of the CARs, the chimeric antigens receptors are the extracellular domain. They have the transmembrane domain and the intercellular signaling domain. So all of them have the different functions. They follow the different procedure and the flowchart where the isolation of the T cells from the patients are done, followed by the genetically modification to experience the chimeric antigens. Then they expand in the culture and reinfuse into the patients. It has certain limitations as well as some future directions. So immunotherapy strategies have the active and the passive approaches. So basically the dendritic cell or the DC vaccination enhances the antigen presentation, where the isolation of the dendritic cells from the patient and for the tumor antigens are used and infused. Then there is the passive immunotherapy where the monoclonal antibodies and adaptive T cells and the immunotoxins are primarily used. This is followed by the immune checkpoint blockades, and it has the different principles, mechanisms, and the clinical applications are discussed here. So the concept and the majority of the inhibitory receptors is that the tumor cells has the inhibitory pathways for the immune cells or the T cells, and they suppress the immune response. The major emitter of the receptors are the antigen presenting cells, which inhibits and reduces the activation. Then the mechanism of the action is by priming the lymph nodes and then effector phase in the tumor microenvironment, then targeting the inhibitor receptors with clinical considerations, and they are the comparison of the checkpoint and the blockade and the vaccination alone. Finally, we look into the optimizing the immunotherapy and the combination and the strategies for the T cell receptor and the modulation. The first one is the combination strategies, which enhances the efficacy, where the therapy of the checkpoint inhibitor as well as the therapy B with vaccine and the kinase inhibitor, for example, enhances the efficacy. There are certain examples and the challenges. The landscape of the T cell receptors is like the balancing the activation and the inhibition. So certain receptors can be activated and certain receptors can be inhibited, and thereby balancing the T cell activation. So the goal is to effective the immunotherapy and requires the modulation of this balance to favor the activation and the over suppression. So with this, we come to concluding the of this course. Whatever is not covered in this lecture will be covered in the subsequent videos or the subsequent tutorials, and we'll be coming up with the different tutorials for analyzing the uh, cancer data sets. We'll be learning about the different databases and other important therapeutic applications of the data sets and their primary case studies and the examples. So to summarize, in this course, we have basically covered the introduction of why to study the cancer. We have discussed about the hallmarks and defined the somatic and the germline and epidemiological basics, how the cancer is studied and from the patient to the population. We have road mapped and the different prerequisites of the cancer progression. Thereby, we have looked at the hallmarks as well as the different molecular approaches of the cancer biology and the cell biology of the cancer, whereas we have looked into the cell cycle control, apoptosis, senescence, and other signaling pathways and other phenomena which are occurring in the cancer. Next, we'll be looking, we have looked into the genetics and the genomics of the cancer, the oncogenes and the tumor suppressors, mutational processes. We have also covered the transcriptional epigenetics and the regulation in the tumors. And we have also looked into the tumor microenvironment with the immune context and the crosstalk between the different immune checkpoints and the metabolic interactions. And we have also covered the cancer diagnostics and the biomarkers in the later parts. Uh, and then we have covered the therapeutics, the targeted drugs, and the immunotherapies like the CAR T and the concepts. The other this tumor evolution, and this also comes under the uh, in the primary lectures. And then we have covered the CAR T conceptuals and resistance mechanisms and all. Then we have looked into the preclinical and the mouse and the animal models. Then we haven't yet discussed the computational cancer biology, which we are yet to discuss. And these 10, 11, 12, and the 14, these are the few uh, lectures which will be completing in the upcoming sessions, because this video has been already been more than 3.5 hours, and so we'll be covering all these things in the upcoming lessons lectures. So keep updated, stay connected with us, and that's it. Thanks and have a good day.